All right. In case you face any lagging issues or you couldn't hear me, please let me know through the chat, or you can DM me through WhatsApp as well. All right, a brief introduction about myself. My name is Ami Kiran and I will be your biology tutor. All right, and in the 2023 LOL exam, I was the district 226 and my island rank was 945. All right, my EZ score was 2.0058 and I got 3 A's. So those are my qualifications. And telling you about the class, from next month onwards, I will be starting my official class. And in that class, I'm planning to do 4 papers, four papers per month. All right, that means I will be discussing 3 MCQ papers, and on the last day, I will discuss an SCQ paper as well as an essay paper. The reason why I'm putting more emphasis on MCQ papers is because you can get 50 out of 50 from that paper easily. I know it's a bit challenging. There are tricky questions, but if you can practice the MCQs for a long time, and if you get the hang of it, like, if you get the hang of it, for example, you do a lot of MCQs and you understand the way it's been coming. For example, from unit 2, there are around like 8 questions. From unit 3, there are around like 4-5 questions. From unit 5, there are like 12-13 questions. And you also know the areas where they usually question. For example, if you take the auxin and the cytokinins, that table in your book, they ask that table from you in almost all the papers. There are some questions, there are some areas in your bio book which always comes for your paper. For example, if you take environmental biology part, they usually ask questions from the endangered species, the vulnerable species, and all those threatened species part. So there are specific areas where they ask the MCQ papers. When you're practicing the model papers, you get a hang of it. You understand, okay, these are the areas which I have to put more emphasis on. Okay. And I will also tell you about, I will also give you more information, like how to write the papers. For example, when it comes to SCQs and essays, there are so many essay questions from your resource book. If you take the 2 resource book, you can't finish, that means you can't finish completely studying it, right? There are so many points in your resource book, but can you just study the guessings that your teacher gives and go to your exam confidently? No, you can't. If you go to your exam with only the guessings in your head, what happens is you lose confidence. But when I went to the paper, I was ready for not only the guessings, but I was also ready for any questions that can come out that can come away from the guessings. For example, unit 1. Do you even think that an essay question can come from unit 1? No, right? No one would ever think that an essay question would come from unit 1. But I saw in a provincial paper that they have asked about an essay from unit 1, issues pertaining to biology. When you get such sort of essay, issue pertaining to biology, would you be able to write that? No, but if you get it for an SCQ, you know what are the different types of, for example, if I take the resource book, you know that due to the overexploitation of natural resources, environmental pollution, loss of biodiversity, desertification can take place. You know those things, right? But if it comes for an essay question, would you be able to organize all those points and write? No, right? That's because you do not think that unit 1 would come for your essay. No one would think that. That's why the essays come from areas where you don't even think sometimes. For example, in the 2023 LOL examination, they told that heart was a guessing, but most of the people studied the structure of the heart and went. Did they study about the angina part, the area where there are cardiovascular diseases and all? They didn't study about those things. So most of the people just study the major guessing and go to the exam. That is not what you should do. You should study everything in the resource book. But the problem is that's not practical. It's not practical to study everything in your resource book. So there are tips and there are techniques to study your resource book. One technique that I used was drawing images. I will tell you more about these things in the essay discussion, which we will be doing from next month onwards. But I'm just going to give you a brief idea. You have to draw what I did was I drew images. For example, cold stress and all the stresses like drought stress and biotic stresses and everything. I drew images to remember the flow of the points, and I studied the flow. That's how I did it. You have to use techniques to study these essays. You can't study all the points. You're not a machine, right? So therefore, techniques are very important in studying essays. Even issues pertaining to biology. I did not study each and every single point in this book. I did not study all the points in this book. What I did was I took out the major points, right? The major points, especially the ones which can be targeted for, which are mostly asked for the CQs. I went through these points several times. I studied it, and I allowed it to go into my head. And later, I was able to write my own essay, and I was able to write it in my own words sometimes, but the point was included. Clearly, the point was included. For example, you can write in your own words, right? The various environmental problems due to overexploitation of natural resources are environmental pollution, loss of biodiversity, desertification. You get 3 points for writing environmental pollution, loss of biodiversity, and desertification. So these are the techniques that you need to implement when writing essays. There isn't anything like that, you have to write the exact wording. That means if there is but, you have to write that word but. There is nothing like that. If the word though is there, you have to write the exact word though. No, you can change the sentence structures, but the main thing is the point should be there. The point should be in your head. And I will tell you more information of how to study microbiology, molecular biology, applied biology, environmental biology after the paper discussion, because my main emphasis on today is on the paper discussion. All right, and I will also tell you about the paper class. That means the paper class that we are going to do from next month onwards. Also, for this paper class, you will take 1 day from your whole month. Can you believe that? You will take only 20 hours. 20 hours from the whole month for this paper class, because I do not have any intention of taking extra time from your day. Going for 2-3 classes during these 3 remaining months is a stupid thing. Okay, you have to stick into one class, but you have to do papers. When I was doing my A-levels, I did not have a proper past paper class. I went for a Sinhala medium class. Okay, that Sinhala teacher was very good. I respect him a lot, but the issue was there was a language barrier. That means there were things that I could understand, but there were things that I had to go home and I had to revise all over again because I couldn't understand some points. So there was a language barrier, and therefore I had to do the past papers alone by myself. During the last 3 months before my A-level exam, what I did was I kept a specific time in the week to revise these past papers, because no matter how many model papers you do, all right, what comes are the techniques that you have to practice gets from the practice from past papers, especially the MCQs, because the flow. I will give you some documents the next day. The classified questions. Now the classified questions, for example, from Unit 1, how many questions came in the 2020 exam? From Unit 2, how many questions came? The ones that I made personally back in those time. So I will share those documents with you. From that you can understand the questions that will be coming for the next paper. And there are some specific questions which are repeated all over again, right, over the years. So I will tell you about the procedure of the class. We take 2 hours 30 minutes for the discussion. Before that, try to do the paper within 1 hour 30 minutes. And at the end, we have to do a review. This reviewing is very important, not only for biology, but also for chemistry and physics. If you are a person who's doing model papers or past papers, you must do reviewing. From reviewing only, you can improve your marks. From reviewing, you understand your mistake and you won't repeat that mistake in your next past paper, in your next model paper. And also make sure you take the model papers seriously. Take it as your final exam and write it. I took all my model papers seriously and I wrote it very seriously, thinking it's my final exam. like assuming that it's exam conditions, sometimes I wrote it online, sometimes I wrote it physically. But I don't think online is a barrier. If you can make your environment, if you can sacrifice anything, you can do that. Okay. All right, then I'll talk about reviewing at the end of the paper discussion. I told you about the class, how the number of hours I'm going to take for the class, and I also told you how to write essays and structured essays. But there are so many other tips that I have to share with you when doing the essays, when doing structured essays, how to write it to the point, and how to get full marks for the SCQs. These are very important things that you need to know. We'll discuss it in our SCQ section. In our SCQ session. Today we are only going to do an MCQ session, and without further ado, let's move on to the discussion. All right, and also I want to say I will stop my video from hereafter because I want to do annotations, and to do annotations, my video will be a barrier for me. So I'm going to stop my video, and if you have any any problems with my network, please let me know through WhatsApp, because I won't be able to see the Zoom chat. Okay, send me a message through WhatsApp. I will check it every 15 minutes to make sure that the meeting is in a smooth condition, right? And I also will share the screen. There is a problem with my past experiences. I have noticed the screen sharing automatically stops. All right, and if it automatically stops in the middle, please let me know, because I don't like to waste time. You have only limited amount of time. You have just 3 months left, and this time is very important for you guys, right? So without further ado, let's move on to the discussion, and I will start sharing the screen from here onwards. Okay, I hope that everyone can see the screen now. Give me a minute. If you can't see the screen, please DM me. Please DM me through WhatsApp, okay? And I will look at it. All right, let's move on to the first question. I hope you all can hear me clearly. If you have any issues, please let me know. All right, first question. They say, select the answer which includes the compounds that have nitrogen as their component. Now, guys, we know usually the first question comes from Unit 1 or Unit 2, right? We know that. First question usually come from Unit 1 or Unit 2. Do we ever think a Unit 4 question would come for the first question? No, right? But there are tricky questions. That's why we need to practice these provincial papers. Okay. If we take the first question, what they're asking is the compounds that have nitrogen. All right, let's go through the first answer. Now, I will tell you how I found the answer while I was doing this paper back in 2023. For most of the question, I used the elimination method. I hope you all know the elimination method. I hope you all know the elimination method. That method is cutting off all the wrong answers and then choosing the correct answer. That helped me to build confidence, because if you can do the first question confidently, you can confidently move on to the second question. And then you do not have to worry about the first question again. But definitely you should again check. That means that you should double check the paper at the end. But what I'm saying is, not to make sure that you do not make careless mistakes in the other questions. You have to make sure that you face this very first question confidently. All right, so that is one important thing that I had to tell you. For that, you have to be thorough with all the points in your resource book. So today I'm going to make sure that you have some CR papers or a book, because I'm going to give you a small review after each and every question, so that you are in touch with the lesson. Your mind won't go here and there when you're in this paper discussion. I want you to write. That's why I want to write. That is not your review, that is my review, okay? After the discussion, you have to write your own review. I will tell you how to do that. You have to take only 30 minutes to do that, okay? All right. Now let's start with the first question. They're asking for the compounds which contain nitrogen. Let's go through the first answer. It says pectin. What is this pectin? Pectin, can you remember in Unit 2, we had this polysaccharide table. In that polysaccharide table, we had pectin, right? And they have given the monosaccharide as galacturonic acid, right? And also they have given some functions as well, but the function is not important. For pectin, it is made out of galacturonic acid. And can you remember in the table that they gave, the only polysaccharide which contained nitrogen was chitin, right? Usually, what are the structural components of polysaccharide? Carbon, hydrogen, and oxygen. Structural components of carbohydrates. Polysaccharides are carbohydrates, right? It doesn't contain nitrogen, but chitin is the only polysaccharide in our syllabus or in the resource book which contains nitrogen. So definitely pectin shouldn't contain nitrogen, right? So you can easily cut off this answer. Why? Because pectin won't contain nitrogen. But let's go through chlorophyll also. Now, as soon as you see chlorophyll, you would get scared. Why? Because chlorophyll, there's nothing called chlorophyll in unit 2. We talked about chloroplast, but did we talk about chlorophyll? We talked about chlorophyll A, chlorophyll B, those things. We talked about the action spectrum and all. But did we talk about the structural components of chlorophyll in unit 2? No, we didn't. We didn't learn the structural components of chlorophyll in unit 2. And then as soon as you see the answer, you'll get scared. How come chlorophyll came into the first question? We didn't even learn about that. You would start thinking like that, right? That is how many children get panicked. You should stop that. You should know all these points. Somewhere in the resource book, they have talked about chlorophyll. I'll come to that. Okay, I'll come to that, but I'll keep it for now. Let's go to insulin. What is insulin? Insulin is a protein, right? Insulin is a protein, and proteins, we know they are made out of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur, right? So therefore, insulin will definitely contain nitrogen. But since pectin doesn't contain nitrogen, we have to cut this answer off, right? And let's go to the second answer. Chitin, chlorophyll, casein. Okay, chitin. Now, I told you before also, chitin is the only polysaccharide which contains nitrogen. So yeah, chitin contains nitrogen. Then this chlorophyll contains nitrogen. Let's keep it. Let's keep chlorophyll for some time. After I discuss everything, I'll go to chlorophyll. Then casein. What is casein? It is the storage protein in milk, right? Storage protein in milk. Now, when I'm saying these things, quickly you should recall these things into your memory. That is what should happen. These are small points. These are also very important. Don't think these are useless points. These are very important points, right? So even though they are easy. So casein contains nitrogen because it's a protein. So this answer is sort of correct, but we still don't know about chlorophyll. So let's keep the second answer and let's go to the third answer. Chitin. Again, they have talked about chitin. Chitin is the only polysaccharide in our resource book which contains nitrogen. So therefore, we can tick it as correct. Then inulin, what is inulin? Inulin is a polysaccharide. It is made up of fructose, right? As you can see in the review that I have given here, inulin is a polysaccharide made up of fructose. Okay, so inulin is also in that polysaccharide table. In the polysaccharide table, we learned chitin is the only polysaccharide which contains nitrogen. That means inulin cannot contain nitrogen. It is a polysaccharide. It contains only carbon, hydrogen, and oxygen. So we can easily cut the third answer as well. But let's go through actin. What is actin? Actin is a motor protein or a contractile protein. In the protein chart, we learn those different types of protein, right? And also we learn about actin in unit 5 in the skeletal system, actin and myosin. So actin is a protein, therefore it contains nitrogen. So we can easily cut off the third answer as well because we know inulin does not contain nitrogen even though actin and chitin contains nitrogen, right? Now let's go for the fourth answer. Myosin, again myosin is a contractile or a motor protein. It contains nitrogen. Okay, then chitin. Chitin is the only polysaccharide in our resource book which contains nitrogen. Then what is glycogen? Glycogen is a polysaccharide made out of glucose subunits, right? It is made out of glucose subunits. Therefore, it shouldn't contain nitrogen. So even though myosin and chitin contains nitrogen, glycogen does not contain nitrogen. You can cut that answer off. Then the fifth answer, immunoglobulin, pectin, and lactose. What is immunoglobulin? It is a defensive protein. Protein means it contains nitrogen, right? So the immunoglobulin is correct. Then what is pectin? Pectin is a polysaccharide. Pectin doesn't contain nitrogen. Therefore, that answer also get cut off because there's pectin. And what is lactose? Lactose, children, it's a disaccharide. Okay, disaccharide. It is made out of what? Galactose and glucose, right? It is made out of galactose and glucose. So since it's a disaccharide, it also doesn't contain nitrogen. And the only remaining answer, even though you didn't know that chlorophyll contains nitrogen, you can circle it as the final answer. But the issue is still something would trouble you because you're not very sure about chlorophyll. How can you be sure about chlorophyll? For that, you have to have studied the resource book perfectly. You have to know all these points in the resource book. Now see, this table is in your resource book, the macronutrient table in unit 4. It is in your resource book. You can see nitrogen is a component of amino acids, proteins, nucleotides, nucleic acids, chlorophyll. See, nitrogen is a component of the chlorophyll. Therefore, we can make sure that the second answer is the final answer and it is the correct answer, right? We can be sure about that. So for question number 1, the answer is the second one. And also we take it from the answers we take it from unit 2 and unit 4. So the question has come from unit 2 and unit 4. It's a combo. Now let's go to the second question. But before that, can I please know, I'm going to check the chat. Can I please know if you understood the first question for me to get an idea? Can I please know whether you understood it? Okay, thank you. Thank you for replying. All right, now let's go into the second question. I have started screen sharing. Okay, I'm starting screen sharing now. All right. Hereafter, I won't ask from after each question. I won't ask you whether you understood. I just wanted to know whether you understood the way I teach you. All right. Now let's go on to the second question. Not a function of lipids. Now, in our resource book, they have given us 4 functions of lipids. All right, but before going through those 4 functions in your resource book, even by your general knowledge, you can answer this question. All right, now how do we use the general knowledge to answer these sort of questions? Look at the answer number 1. Food reserve as an energy source. We know that after all our carbohydrates gets finished up, what happens is, after all our carbohydrates finishes up, what happens is lipids act as the energy source, right? So therefore, the lipid reserve or the fat reserve in our body definitely act as an energy source when we do not have enough energy inside our body, right? When we do not have instant energy inside our body, what happens is the lipid reserve or the fat reserves in our body, the fat layers under the skin gets used up or in the liver gets used up. For what? As a quick energy source, right? So number 1 is definitely correct. From your general knowledge, you can answer. But remember, these points are given in your resource book, okay? All right, now let's go to the second one. Maintain the fluidity of the plasma membrane. What are the lipids that maintain the fluidity of the plasma membrane? They are the cholesterol molecules and the phospholipids. Now we know the phospholipid bilayer is arranged like this. And inside there are cholesterol molecules. So both these molecules provide the fluidity of the plasma membrane. That's why we call this phospholipid structure as the fluid mosaic structure. Why do we call it fluid mosaic? Because these phospholipids are always moving. That's why we call it fluid. And why do we call it mosaic? Because protein molecules are integrated. That is out of the question, but I just wanted to tell you the fluid mosaic theory as well. So somehow these phospholipids as well as the cholesterol molecules maintain the fluidity. Now, how does this cholesterol molecules maintain the fluidity? I will go out of the question because I want to revise all the points. That's why I'm going out of the question. How does the cholesterol molecules? We learned that under cold stress, right? We learned that under cold stress in unit 4. Cold stress, we learned when the temperatures are high, it makes sure that the fluidity of the plasma membrane is less. And when the temperatures are low, it makes sure the fluidity of the plasma membrane is high. That's how it maintains the fluidity. When there are low temperatures, it won't allow the plasma membrane to get frozen. It allows some fluidity. That's the importance of cholesterol molecules. And when the temperature is hot, it doesn't allow the plasma membrane to be too fluid. It allows some stability in the plasma membrane, right? So that is the you should know these things in general knowledge. You don't have to memorize everything. To memorize also, you should know these basics, right? First, know your basics. Especially these techniques are very important in the molecular biology part. You can't study everything in molecular biology. You need different techniques to study molecular biology. I will come to that. But let's go to the question now. Now we know maintain the fluidity of plasma membrane is correct, right? Active signaling molecules that travel through the body. What are these signaling molecules called as? These signaling molecules are called as hormones. But over here, I told you insulin, we know it's a hormone, but it's a protein, right? But lipids, now there are some lipids also act as hormones. There is a problem. No, there's no problem like that because hormones can be both lipids and proteins, right? What do we call here? It's there. Sorry, yeah, here it's there. What do we call the lipid hormones? We call the lipid hormones as the steroid hormones. All right, estrogen, testosterone, these are steroid hormones. And what are the protein hormones? There are so many protein hormones in our body. Insulin, glycogen, those are protein hormones. But estrogen, testosterone, and some sex hormones are actually lipid hormones. And they are called as steroid hormones. All right, so the third one is also correct. They are basically referring to the what? Signaling molecules means the hormones, right? So the third one is also correct. Fourth one, participating in cell identification. Now the cell is there, the cell membrane, we know they have protein molecules, right? These protein molecules are the ones which are involved in cell identification. If you have read the plasma membrane part carefully, under the plasma membrane, they have told you about the functions of the proteins in the plasma membrane. In that, cell identification is one of the functions, right? So you should know it's not a function of the lipids, it's a function of the proteins embedded in the plasma membrane. So the wrong answer is, the answer is actually 4. Okay, the answer is the 4th one, but let's go through the 5th one as well. Found as components of cell membrane. What are the components of cell membrane? What are the lipid components of cell membrane? Phospholipids. Number 1 is phospholipids. Those are also type of lipids, right? Phospholipids is a type of lipid. So it is a component of the cell membrane. What is the other lipid component of the cell membrane? Cholesterol molecules. Both these molecules are components of the cell membrane, as well as both these molecules maintains the fluidity of the plasma membrane. So from that, we can easily get the answer as the 4th answer. I hope you all understood that. With that, is the screen being shared? I hope the screen is being shared. All right. If there is any issue, please let me know through WhatsApp. I'm checking WhatsApp. All right. Okay. Now let's go through the 3rd one. What is incorrect about plasma membrane? Now, in the 2nd question also we discussed about plasma membrane. So I will go through this question very fast, but let's discuss all the points. Number 1, mainly composed of phospholipid and protein molecules. Yes, we know that the plasma membrane is made out of phospholipids like this, phospholipid bilayer actually. You can see there is a phospholipid bilayer, and in between there are protein molecules embedded, right? Called integral proteins, right? All right, therefore the first one is correct, no doubt about that. Now let's go to the second one. The hydrophobic head of the phospholipid molecules faces inwards. What do you mean by hydrophobic? Now, acrophobic means scared of spiders, right? What do you mean by hydrophobic? Phobic means also scared. Scared of what? Scared of water. So hydrophobic means that part is scared of water. That part won't face any area which has water. It repels water, right? So the head molecules, they're saying they are hydrophobic. Are they hydrophobic? Can you see these head molecules are facing outwards to the external environment of the cell? In the external environment, there are so many fluids, and also these head molecules are facing to the internal environment of the cell. And the internal environment also, there are so many fluids. In these fluids, definitely water is there, right? Water is the main constituent in this fluid. Therefore, can you say these heads are hydrophobic? Are these heads hydrophobic? No. These heads are hydrophilic. These tails are the ones which do not like water. Can you see? The tails do not like water. That's why they face inwards, right? That's why they face inwards. Because the tails do not like water, and those are called the hydrophobic tails. All right. So I hope you understood the difference between the hydrophobic and hydrophilic, and also you understood why the tails face inwards and why the heads face outwards into the external and the internal environment. So from that itself, we can understand the second one is wrong. Why? Because the hydrophobic head of phospholipid molecules, it's not the head, it's the tail. The hydrophobic tail of the phospholipid molecules faces inwards and creates a hydrophobic interior. All right, so it's a hydrophobic tail actually. I'm sorry I couldn't give you a review to the first and second question, right? Shall we write down a review for the first and second question? If you can, please take a writing book or please take some CR papers. I'm really sorry I forgot to give you a review. Shall we write down the review for the first one? All right, now I will start reading the review. For the first one, write down chitin is a polysaccharide. Chitin is a polysaccharide which contains nitrogen. Chitin is a polysaccharide which contains nitrogen. Okay, and please let me know if I'm fast. Chitin is a polysaccharide which contains nitrogen. That's what I said. And then write pectin, inulin, glycogen are polysaccharides. Pectin. I said chitin is a polysaccharide that contains nitrogen. Okay. And now I'm saying pectin, inulin, and glycogen are polysaccharides that contain only carbon, hydrogen, and oxygen. Pectin, inulin, and glycogen are polysaccharides that contains only carbon, hydrogen, and oxygen. And you can write chlorophyll contains nitrogen. Chlorophyll contains nitrogen. That is the review for question number 1. Now I will give you the review for question number 2. Okay. Question number 2, write down the review. Write down the review. You can write down this review actually. There are 4 functions of lipids given under unit 2. There are 4 functions of lipids given under unit 2. Lipid hormones are called steroid hormones. Lipid hormones are called steroid hormones. These lipid hormones are called steroid hormones. Okay. And that is not in your resource book. That is for your general knowledge. You should know these things. Then only the flow of the theory comes into your head while doing the papers. All right. Estrogen and testosterone are steroid hormones. Estrogen and testosterone are steroid hormones. All right. You can write down that in the review. Give me 1 minute. All right. So I hope that everyone wrote that review. Now let's go back into the discussion. All right. So we stopped in the second one. We understood that the second statement is a wrong statement. Now let's go into the third statement. Phospholipids provide fluid nature to the membrane as it has a movable nature. Yes, we know these phospholipid molecules move up and down because sometimes to allow the transport of materials. I wish that I could show you an animation. Actually, when you are learning Unit 1 and Unit 2, you should put more emphasis on to seeing these animations. Then only it goes into head. But now you guys don't have time to see these animations. But somehow these phospholipid molecules move up and down to allow transportation of substances. There are what you call channel proteins as well, transmembrane proteins, which allow the what you call passage of different substances into the cell. But anyhow, these plasma membranes has a fluid nature. They vibrate, they do not vibrate, but somehow they move up and down and they have different fluid natures which can be shown by animations. All right, after your A-levels, you can watch those things. But for now, please understand that phospholipids have a fluid nature to the membrane. All right, so that one is correct. And the fourth answer, it says glycoproteins have short branching carbohydrate chains like antennae. Now, children, in the phospholipids, like this, in the phospholipid bilayer, there are protein molecules also integrated like this. They're called as integral proteins. There can be carbohydrate chains like this. These are used for actually cell identification, and also, yeah, these are mainly used for cell identification, right? And you can see over here, these protein molecules also have carbohydrate chains. When a carbohydrate chain is attached to a protein molecule, we call it glycoprotein. We call it glycoprotein. And when a carbohydrate chain is attached to a phospholipid, we call it glycolipids, right? That's how these words are formed. Okay, so glycoproteins have short branching carb. Now these are short, these are not very long, right? These are short branching chains like antennae. These are like antennae, right? So therefore the fourth answer is correct. Now let's go to the fifth answer. Fifth answer says the two sides of the membrane may differ in composition and function. That is a statement given in your resource book. If you check your resource book, if you have studied your resource book, definitely you must have seen this sentence. Okay, so the two sides of the membrane differ in composition. This side differ in composition. Now this is the outer side, this is the inner side. This side differs from composition and this side differs from composition as well as function, right? So you need to understand those things. Those are in your resource book. Now, write down the review for that. Write down the review for the third question. Some proteins and lipids have short branching carbohydrate chains. Some proteins and lipids have short branching carbohydrate chains like antennae. Some proteins and lipids have short branching carbohydrate chains like antennae, forming glycoproteins and glycolipids respectively. Some proteins and lipids have short branching carbohydrate chains like antennae, forming glycoproteins and glycolipids respectively. All right. I'm repeating it once again. Some proteins and lipids have short branching carbohydrate chains like antennae, forming glycoproteins and glycolipids respectively. So that is the review for the third one. All right, now let's go for the fourth one. They're talking about lysozymes. Lysozymes are single membrane bounded vesicles. That is true. Because we learn in our what you call second unit itself, I think at the not at the start, but I think when we are learning about the animal cells and all, in that table we learn the ones which are surrounded by single membranes and ones which are surrounded by double membranes. As a revision, I'll tell you all those things now. What are the structures in a cell which are covered by a single membrane? Lysozymes and vacuoles, right? Lysozymes and vacuoles are covered by single membrane. Then what are the structures which are covered by double membrane? Nucleus, mitochondria, chloroplast. Now you will wonder, what about ribosomes? Ribosomes are also structures in the cell, right? But they are not organelles. They are subcellular structures, right? Ribosomes are subcellular structures. Why do we call them subcellular structures but not organelles? Because they do not have a membrane. They are not covered with the membrane. That's the reason why we do not call them as organelles, and that's why we call them as subcellular structures, right? All these are also subcellular structures, but these are also called as organelles. But this is just a subcellular structure, not an organelle. Organelles are covered by membranes, right? So I hope you understood that point. Those are simple points that we learned when we are studying Unit 2 and Unit 1. Okay. All right. Now, they're single membrane bounded vesicles. We have no doubt about that, right? So it has been proved they're single membrane bounded vesicles. Now let's go for the second one. They contain hydrolytic enzymes. Do they contain hydrolytic enzymes? Yes. What do you mean by hydrolytic enzymes? Enzymes which promotes hydrolysis, right? Hydrolysis. That means the breakdown, it catalyzes the breakdown of carbohydrates, of proteins, of lipids, of nucleic acids. Okay, lysozymes catalyzes the breakdown of these 4 organic molecules that we have studied in Unit 2. We have studied about carbohydrates, we have studied about proteins, we have studied about lipids and nucleic acids, right? So it contains hydrolytic enzymes. And what is the organelle which contains oxidizing enzymes then? Oxidizing enzymes are contained in peroxisomes. Okay, the difference between one difference between peroxisome and lysosome is the is the content inside that. For example, lysozymes have hydrolytic enzymes and peroxisomes have oxidizing enzymes. But lysozymes, one of the major differences, lysozymes are found only in animals. They are found only in animals. But if you take peroxisomes, they are found in both plants and animals. That is one major difference between these two vesicles, because both are vesicles, right? So you might get confused with lysozymes and peroxisome. That's why I mentioned that difference. Okay, so they contain hydrolytic enzyme. That answer is correct. They help to digest worn out particles. Children, under lysosomes, there are 4 points given, 4 functions given to us. What are the 4 functions? They digest food particles received by phagocytosis. Okay, they digest food particles. What do you mean by phagocytosis? Engulfing food particles. This process is called phagocytosis, right? If you have heard about phagocytes, the process that they use to engulf the pathogens is what you call phagocytosis. So we basically use this to explain the process of engulfing phagocytosis. So they digest food particles received by phagocytosis. That is the number 1 function. Now, what is the second function? They transport. Now, after you get in all these food materials, you digest all these food materials, and there are residue and waste substances inside this cell. Now, how do you remove them out of the cell? You have to remove them by a process called exocytosis. So the second function is they transport the residue material out of the cell by exocytosis. That is the second function, right? The third function is they digest worn out organelles, right? Inside the cell, if the chloroplast or the mitochondria is old, they can digest these organelles, right? So they digest worn out organelles. Therefore, the third answer will be correct. And the fourth answer is also correct because I told you, sorry, fourth answer is not correct. Fourth answer is not correct. Second and third is correct. Now see, I also, as soon as I saw the word phagocytosis, I made a mistake. I was in a hurry and I ticked it as correct. But no, it is the wrong answer. Let's see why is it the wrong answer. Helps in transport of food particles received by phagocytosis out of the cell. No, they transport these residue materials, or they transport this what you call food particles out of the cell by the process called exocytosis, not by phagocytosis. Can you see? You make careless mistakes. I'm also a student, right? I just finished my A-level exam. See, I also made a mistake as soon as I saw the word phagocytosis. That is the reason why we should be very alert when we are doing these papers. We should go through each and every word in these sentences, because by one word, the statement can be made wrong. How is this statement made wrong over here? By the word out of. If they say into, then it is correct, right? But they have said out of the cell. Out of the cell, what is the process? Exocytosis. Into the cell only, it is called phagocytosis, right? Now, the fifth one is autolysis causing cell death. We all know that is one of the famous function of the lysosome. It kills itself. That means when it reaches a certain age, it kills itself by digesting all the worn out organelles inside itself, and it goes into and it goes into the phase of death, right? Okay, so we've discussed 4 questions now. I will give you the review for the fourth one. I think, yes, write down this review. I'm going to give the exact same review of, no, I'm going to give you a different review. I'll give you a different review. Lysozymes are single membrane bounded vesicles. Lysozymes are single membrane bounded vesicles present only in animals. Lysozymes are single membrane bounded vesicles present only in animals. Lysozymes are single membrane bounded vesicles present only in animals. Okay. And lysozymes contribute to digestive activity. Lysozymes, or you can write, they contribute to digestive activity. They contribute to digestive activity. You can write the third point as well. They catalyze the breakdown of carbohydrates, proteins, lipids, and nucleic acids. All right. They catalyze the breakdown of carbohydrates, lipids, proteins, and nucleic acids. Right, so that is the review for the fourth question. If I'm very fast, please let me know. I will reduce my speed. All right, now let's go to the fifth question. Cytoskeleton. Now this seems like a boring part in Unit 2, but you have to love the content in your resource book. When you love the content, you start memorizing these things very easily. That's what I did. I also started loving this content. This is very beautiful. Okay, now let's go to cytoskeleton. What do you mean? What is this cytoskeleton? What is the function of cytoskeleton? We know cytoskeleton is the supporting structure of the cell. These cells have a supporting structure, just like we have a skeleton. These cells also have a skeleton inside that called the cytoskeleton. You know, plant cells have a cell wall, right? They have a cell wall to maintain its structure. But we, animal cells, they do not contain a cell wall. Because of that, just imagine if they did not contain a support, our cells would run here and there because it's just a fluid, right? It won't have any perfect structure. It won't have, that means perfect structure in the sense, it would be just a whole mass of fluid, right? If we didn't have a cytoskeleton. So that is the reason why we need to have a cytoskeleton in our cells, because it is the supporting structure of the cell and it maintains its shape. Now, what is the cytoskeleton made up of? okay, I'm not going to ask questions, but the cytoskeleton is made out of three things, right? Three components. What are those components? The smallest component is the microfilaments, right? In your syllabus, you do not have to learn the, what you call, the big table that you got. But in our syllabus, that means after 2024, you got a new book in the sense some amendments were made in that book. So you do not have to study that table, but we had to study that table, okay? So it's better if you know the content in that table, even though you didn't memorize it. Knowing the content helps you a lot in MCQs. All right. So microfilaments, also called as actin filaments, are the smallest. And then comes the intermediate filaments. That's why they call it intermediate, because it's in the middle. That is the second smallest. Sorry, this is the smallest. Then this is larger than the microfilaments. Then the biggest is the microtubules, right? So this is the order: microfilaments, intermediate filaments, and microtubules. These are the three components of the cytoskeleton. I hope that all of you know the three components, and also you need to know the functions of the cytoskeleton. All these functions that they have given, guys, all these things over here, they are exactly in your resource book. The thing is, we have to study the resource book. Keep on, when you're studying now, you have a particular time in your week, right, to study biology. You have divided your week into some different, you have a timetable, right, to study physics, to study biology, because you need to balance the three subjects during the last 3 months, right? So you need to have a time for biology, and when you're studying the biology, it's okay. Even if you do know the theory part well, keep on rereading those if you had time. If you are traveling, keep on rereading those. And one technique that I used was I made my own recordings, own voice recordings, and whenever I was traveling, I we used to listen to songs when we are traveling, right? But I changed that trend. What I did was I used my earphones to listen to the recordings while I'm traveling. It was all biology in my head when I'm traveling. Whenever I'm going to a class, whenever I'm coming out from class, if I'm going to the hospital, if I'm going to any event or anything like that during my A-level times, what I did was I listened to the recordings that I made, and I will share those recordings with you guys as well, because now you guys don't have time to make recordings for yourself, right? But I also made it during the last 3 months, to be frank with you. Right? Okay. So the fifth one, provides strength to the cytoplasm. Yes, the cytoskeleton provides enough strength to the cytoplasm, right? That's why it maintains its shape, right? Anchorage organelles. Just imagine if these organelles were not placed in one place, if they were not anchored in one place, one day it would be over here. One day the nucleus would be over here. Right? So there's going to be a lot of problems if these organelles are not anchored in one place. They are moving, okay? They are not like, perfectly still. They are not in that perfectly stationary position. They do move, but their movement is controlled by this anchoring. Otherwise, many problems can occur inside the cell, right? Collision of organelles and all those things. So anchoraging organelles is another function of the cytoskeleton. Movement of the cytoplasm. Okay, movement of the cytoplasm is actually done by the microfilaments. If you know the table, even if you haven't memorized it, microfilaments are the one which is responsible for the movement of cytoplasm. There is this thing called cytoplasmic streaming. Why does the cytoplasm stream? Because there are so many enzymes, right? The enzymes need to be carried into some specific places inside the cell, right? And for transportation of specific proteins, amino acids inside the cell, there should be some movement, right? Does the amino acids have a brain to travel by its own? No, right? It should be, carried by something. It is carried by the cytoplasmic stream. So movement of cytoplasm is a function of the cytoskeleton. And maintaining the shape of the cell is definitely one function of the cytoskeleton. And limiting the growth of cell is not a function. Have you ever heard that because of the cytoskeleton, the animal cell can't grow? We grow, right? Our cells grow, right? Our cell starts from an early stage and goes into the mature stage and even die after that. Because why? The cytoskeletons won't limit our cell growth, right? So you need to know these specific general points. They are not in the resource book, right? They haven't said that the cytoskeleton does not limit the growth of the cell, but you have to know it won't limit the growth of the cell, right? So these are some general things that you need to know. All right. Now let's go. Okay, I didn't give you a review for the fifth one. Now let's write a review for the fifth one. Cytoskeleton. Cytoskeleton is the supporting structure. Cytoskeleton is the supporting structure of the cell and maintains its shape. Cytoskeleton is the supporting structure of the cell and maintains its shape. All right. And write the next point. There are three types of components in the cytoskeleton. There are three components. There are three types of components in the cytoskeleton. They are, right in order from smallest to largest, microfilaments or actin filaments. You call microfilaments actin filaments. Microfilaments or actin filaments. Microfilaments or actin filaments. And then write, I'm looking at your messages, that's why I'm taking some time. You can go through microfilaments or actin filaments. That is the first one. The second one is intermediate filaments. Intermediate filaments. All right. The third one is the microtubules. Microtubules. All right. So I hope you have written that review. I will repeat it again. Once, cytoskeleton is the supporting structure of the cell and maintains its shape, right? There are three types of components in the cytoskeleton. They are microfilaments or actin filaments, intermediate filaments, then microtubules. I gave you it in a specific order from smallest to largest, right? Okay, without further ado, let's move on to the other questions as well. Sixth one, act as coenzyme, act as electron carrier, act as oxidizing agent. As soon as you see these three things, even without looking at the question, the thing that should come into your head is about the nucleotides. Apart from ATP, we learn about some nucleotides. They are NAD+, NADP+, and FAD. Guys, in our syllabus, FAD, they didn't give FAD, under FAD, they didn't give oxidizing agent as a function in our syllabus. And that means in our book. But in your book, they have given, I think they gave, sorry. They gave oxidizing agent, but they didn't give something in our resource book. Let me check it. Yeah, they didn't give FAD and FAD in my book. They didn't give. I think in the later after some other publications they might have written oxidizing agent, because this was a 2023 book, right? This is a 2023 paper, and they have given fifth one as the answer. So therefore, definitely, it was in one of our other books. But somehow in your book, they have given FAD, under FAD, they have given that it acts as an oxidizing agent, right? And all these three molecules, remember, NAD+, NADP+, FAD, act as a coenzyme, act as an electron carrier, act as an oxidizing agent. And you should also know in which process this NAD+ act as an oxidizing agent. Okay, NAD+ act as an oxidizing agent in respiration. We have learned in respiration, NAD+ is the oxidizing agent. It gets reduced and it gets converted into NADH. Children, what do you mean by oxidizing agent? Oxidizing agent, we learn in chemistry also, is an agent or is a compound which gets reduced. That means it itself gets reduced and it makes the other compound oxidize and it gets reduced. Remember, children, when something gets reduced, a hydrogen, not in all the cases, but in most of the cases, when something gets reduced, a hydrogen gets attached to it. So when NAD plus gets reduced, what happens is a hydrogen gets attached to it and it gets converted into NADH, right? NAD plus gets converted into NADH. That means NAD plus has got reduced. It has acted as an oxidizing agent and it has got reduced. When something acts as an oxidizing agent, I'm repeating, that compound gets reduced. The one which is acting as the oxidizing agent gets reduced and the other compound is the one which gets oxidized, right? For example, if you take the anaerobic respiration and all, in anaerobic respiration, we know that NAD plus gets converted to NADH, right? For example, in ethyl alcohol respiration, ethyl alcohol fermentation, we know that NAD plus gets converted to NADH, and the final hydrogen acceptor is the acetaldehyde. Okay, I'm not going to confuse you with those things, but somehow for the moment you should remember when NAD plus gets reduced, it gets converted into NADH. Usually the hydrogen gets connected when something gets reduced, right? So that is what you should know. And I think you got a bit confused with the ethyl alcohol fermentation. We'll come to ethyl alcohol fermentation in the 7th question. So stop thinking about that if you got confused in that area for now. I will come to that. I will clear your doubts very soon. Right. So where do you think NAD plus acts as an oxidizing agent? NAD plus acts as an oxidizing agent in photosynthesis. After accepting electrons, NADP plus gets converted into NADPH. That means this substance oxidizes something else and it gets reduced to NADPH. That's why I said in most of the reduction, a hydrogen joins. When something gets reduces, usually a hydrogen joins. All right. If you didn't know, I will tell you something. I think it's out of the syllabus also, but I will just tell you this is a general knowledge. During reduction, electrons are accepted and hydrogen is accepted. Sometimes there can be electrons accepted and sometimes there can be hydrogen accepted, right? There isn't a must that both should be accepted, either one of them, right? So in this case, both electrons and hydrogen are accepted, right? So that is how reduction takes place. And FAD, guys, they haven't given you a specific function. That means FAD, they haven't given you in which process it acts as an oxidizing agent, but we know FAD is there in photosynthesis, right? So not in photosynthesis, in aerobic respiration, right? In the I think in the what you call the Krebs cycle, we learned that, right? Right. All right. Now I will give you a small review for the 6th question. If you have any doubts regarding that, please let me know. Please ask me through WhatsApp. I will answer you. Because this part is not confusing at all, but because I included that hydrogen part into that case, I have seen some of my peers, like, while I was doing it, it was a bit confusing for people who was in my class at that time. So that's why I thought that it would also be confusing for you guys. That's why I explained it. Anyhow, if you've got any doubts, please let me know. All right. And you can write the review. The answer is the 5th one. We all know that all these things, NAD plus, NADP plus, and FAD act as coenzyme, electron carrier, and oxidizing agent, right? Now you can write the review. NAD plus, NADP plus, and FAD, NAD plus, NADP plus, and FAD act as a coenzyme, act as an electron carrier, act as an oxidizing agent. That is a small review for the 6th one. All right, children, I think in the second one, in the lysozyme part, if I mentioned lysozymes instead of lysosomes, I'm sorry. Lysozymes is something else which we learn under immunity. Lysosomes is something else, okay? Here, lysosomes are the, what do you call, organelles, and lysozymes are actually an enzyme, okay? I think I mentioned it by mistake while I was rereading the answers and everything. So I'm sorry if I got you confused, okay? All right, guys. Now, since you have written the review for the sixth question, I will move on to one of my favorite questions. Seventh questions. I really like this ethyl alcohol fermentation and lactic acid fermentation part. It's very interesting. Okay, now children, both in ethyl alcohol fermentation as well as in lactic acid fermentation, the first step is glycolysis, right? In glycolysis, children, what happens? One glucose molecule gets converted into two pyruvate molecules. One glucose molecule gets converted into two pyruvate molecules, and this releases two ATP molecules. I'm talking about the net ATP production, 2 ATP, all right? And it gives two molecules of NADH, right? It gives two molecules of NADH. I'm talking about the net production in ATP. Actually, 4 molecules of ATP are produced, but initially we use 2 molecules for the breakdown of glucose, and therefore that's why we call the net production of ATP is 2. So glycolysis is the process of breaking down glucose into 2 pyruvate molecules, into 2 ATP molecules, and to 2 NADH. I think they have itself said the first step is glycolysis in ethyl alcohol fermentation. That is true. Where does glycolysis takes place? Glycolysis takes place in the cytosol of the cell. All the enzymes required for glycolysis are in the cytosol of the cell, right? So it doesn't happen in any subcellular structure or in any organelles. It happens in the cytosol. Now let's go to the B part. One molecule of glucose is converted to see exactly the thing I said. It's in your resource book as well. One molecule of glucose is converted to 2 pyruvate, 2 ATP, and 2 Na. But see, not NADPH. It's NADH. All right, not NADPH. It's NADH. The answer is NADH. Therefore, the second one is wrong. By a single letter, that answer could go wrong. That is why I said you have to make sure when you are reading the answers, you are reading every part mindfully. Mindfulness is very important when you are doing a paper. If your mind goes here and there, first you have to practice writing papers. You have to sit, you have to time, and then you have to do the papers by concentrating only on the paper. If there are questions that you don't know, skip those questions. Do the questions that you know mindfully so you do not make careless mistakes. That is one major tip when you are doing papers. Okay. All right. Now the first A is correct, B is wrong. C, carbon dioxide releases during the conversion of pyruvate into ethyl alcohol. I will explain you the process of ethyl alcohol beautifully. So in ethyl alcohol, children, in ethyl alcohol, what happens is first step is glycolysis, right? What does glycolysis produce? Glycolysis produces 2 pyruvate molecules, 2 ATP, and 2 NADH. In ethyl alcohol, this pyruvate is involved in 2 steps. Now, I told you glycolysis produces 2 pyruvate molecules, right? So, this pyruvate molecule is involved in 2 steps. In the first step, pyruvate is converted to acetaldehyde. Acetaldehyde, right? Acetaldehyde. And this process releases a carbon dioxide molecule. All right, because this is a 3 carbon molecule and this is a 2 carbon molecule. So one carbon dioxide should be given, one carbon molecule should be given off, and that carbon molecule is given off in the form of carbon dioxide. So pyruvate is converted to acetaldehyde, right? And in the second step, acetaldehyde, now I told you pyruvate is involved in two steps, right? In the first step, pyruvate is converted to acetaldehyde. In the second step, pyruvate, what happened to this acetaldehyde is reduced to ethanol. It is reduced to ethanol using NADH. Using NADH, children, not NAD plus, NADH. All right, that means this is already the reduced form of NAD plus. NAD plus is the oxidized form. After it gets reduced only, you call it the NADH, right? So NADH, acetaldehyde gets reduced to ethanol in NADH. Acetaldehyde gets reduced to ethanol using NADH. That means NADH gets oxidized to NAD plus. I told you what do you mean? I told you in reduction we take a hydrogen, right? That means in oxidation we remove a hydrogen. Can you see? NADH has got oxidized to NAD plus. It has removed a hydrogen, and this acetaldehyde has gone to what? Acetaldehyde has reduced itself to ethanol, right? So that is a simple process that you need to understand. This reduction process is not that hard, okay, children? NADH, after removing an hydrogen, it gets oxidized to NAD plus. If acetal gets reduced, this is a redox reaction. If one gets reduced, one should get oxidized, right? So NADH has got oxidized to NAD plus, right? So what is the final hydrogen accept in this case? Who accept this hydrogen? Now, NADH removes the hydrogen and go to NAD plus, right? Then who accepts this hydrogen? Acetaldehyde accepts the hydrogen. Acetaldehyde accepts the hydrogen and it gets reduced. I told you when a compound accepts a hydrogen molecule, it gets reduced. So acetaldehyde gets the H from the NADH. It accepts the hydrogen molecule and it gets reduced to ethanol. So did you understand the process of ethyl alcohol? I'll give you a summary of ethyl alcohol. First step is glycolysis. Guys, in glycolysis, 2 molecules of pyruvate, 2 molecules of ATP, 2 molecules of NADH is produced. All are 2. Just remember, 3 compounds are produced and all these 3 are produced in groups of 2, right? Not in groups of 2, numbers of 2, right? In numbers of 2. And the pyruvate is involved in 2 steps. In the first step, pyruvate is converted to acetaldehyde. Pyruvate is a 3-carbon molecule. Pyruvate is a 3-carbon molecule. Acetaldehyde is a 2-carbon molecule. Pyruvate is converted to acetaldehyde by releasing carbon dioxide. And then in the second step, acetaldehyde is reduced to ethanol. What do you mean by reduced? Taking a hydrogen. So acetaldehyde has taken up an hydrogen and got reduced to ethanol. Where did the acetaldehyde get the hydrogen from? Acetaldehyde got the hydrogen from NADH. And NADH therefore got oxidized to NAD plus, right? NAD plus is being recycled over and over again in the ethyl alcohol fermentation process because chemical substances are limited in ethyl alcohol fermentation process, right? I think we learned that under anaerobic respiration. So I hope that I've clearly explained you the process of ethyl alcohol fermentation, right? Okay. If you have any doubts, ask me through WhatsApp, okay? I will check the WhatsApp in after doing this question. Carbon dioxide releases during the conversion of pyruvate into acetaldehyde. Now you know the answer, right? Now you have confidence. Since you have revised it with me, now you know pyruvate, when you turn pyruvate to acetaldehyde, carbon dioxide is released. So A is correct, C is correct. Now let's go to the oxidized into ethanol. Was that what I was explaining all this time? No, right? I told you acetaldehyde is reduced to ethanol, right? Acetaldehyde takes up a H and gets reduced. So therefore, D is wrong. E, they say the final hydrogen acceptor is acetaldehyde. We have no doubt about that. That's what we will be learning all this time, right? The final hydrogen acceptor is acetaldehyde. Therefore, the correct answer is the third one. All right, now I will check your messages. If you've got any messages, please please send me now, because now I will be going through your messages. Through WhatsApp, please. Okay. I think no one has questions. Now I will give you a small review for the 7th one. Small review for the 7th question. Actually, it's not a small review. It's going to be a bit of a big review, because anyways, I'm going to give it to you. Right, ethyl alcohol. Right, like this. I will give you a small table. This won't waste time. You will learn something. That's why I'm going to give you this. Ethyl alcohol in one side. And write lactic acid fermentation in the other side. I'm just going to show you the differences between ethyl alcohol and similarities and differences in ethyl alcohol and lactic acid. Okay. Right here, write as the first point, put an arrow, write like this. One molecule of glucose, you can include it into the lactic acid round as well, because in both lactic acid and ethyl alcohol, glycolysis is the first step. Right, so I'm talking about glycolysis. Right, right, if you have already written, just keep it, but if you have not written yet, write down from the beginning. In glycolysis, one molecule of glucose, is converted to 2 molecules of pyruvate. 1 molecule of glucose is converted to 2 molecules of pyruvate. 1 molecule of glucose is converted to 2 molecules of pyruvate, giving 2 molecules of ATP, and 2 molecules of NADH, and giving 2 molecules of ATP and giving 2 molecules of NADH. Right. I will repeat that part again. I hope my screen is shared once again. In this part, right, in glycolysis, 1 molecule of glucose is converted to 2 molecules of pyruvate, giving 2 molecules of ATP and 2 molecules of NADH. Glycolysis is common for both ethyl alcohol and lactic acid fermentation. But in the second one, just write it under ethyl alcohol. Don't write it under lactic acid. Right? Pyruvate is involved in 2 steps. Pyruvate is involved in 2 steps. Right? Pyruvate is involved, just write on the ethyl alcohol, this part. Pyruvate is involved in 2 steps. Pyruvate is involved in 2 steps. In the first step, pyruvate is converted to acetaldehyde, releasing carbon dioxide. In the first step, pyruvate is converted to acetaldehyde, releasing carbon dioxide. In the first step, pyruvate is converted to acetaldehyde, releasing carbon dioxide. Pyruvate is involved in 2 steps. In the first step, pyruvate is converted to acetaldehyde, releasing carbon dioxide. In the second step, acetaldehyde is reduced. In the second step, acetaldehyde is reduced to ethanol using NADH. In the second step, acetaldehyde is reduced to ethanol using NADH. In the second step, acetaldehyde is reduced to ethanol using NADH. Right. In the lactic acid part, right now, put the second arrow, put the second star and write pyruvate is directly reduced by NADH to lactic acid. I'm writing it down, you can copy it. Sorry for the handwriting. Pyruvate is directly reduced by NADH to lactic acid. All right, so here can you see the difference? In ethyl alcohol, after glycolysis, pyruvate is involved in 2 steps. But here, there are no 2 steps in lactic acid. Pyruvate gets directly reduced to lactic acid, right? In lactic acid, pyruvate gets reduced directly to lactic acid. All right, now you can separate it with the line or something. Now I'm going to the third point. Third point, under ethyl alcohol, final hydrogen acceptor is acetaldehyde. Final hydrogen acceptor is acetaldehyde. And in the second one, you can write final hydrogen acceptor is pyruvate. In the lactic acid part, write final hydrogen acceptor is pyruvate. I'm giving you this table because when you look at the resource book, it seems like a big headache. You have to solve these things by yourself. But now I have separated these into 2 parts so you know the exact difference between these two. And you can study these points and write your own essay, right? And you can even include this in your structured essay. And you can even take these points and then use it for your MCQs, right? So that is why I tried my best to make it easy for you. Take the resource book points and then give it to you in a manner which makes it easier for you to study, right? All right, and then you can write the 4th star. This is common for both. In both, NAD plus is synthesized. NAD plus is synthesized because we know NADH gets oxidized to NAD plus, right? In both, NAD plus is synthesized, right? So I hope that is clear for you. I hope everyone has written that down. If I was too fast, let me know. I can repeat anything if you want. Send it over WhatsApp, please. I'm checking WhatsApp, that's fine. It's hard for me to check the Zoom chats while I'm sharing the screen. Okay. Now, the 7th question review is over. Now let's go to phylum Lycophyta. Lycophyta is also one of my favorite parts, right? Okay, so in phylum Lycophyta, I want to tell you, actually, these are called lycophytes, right? Lycophytes. These lycophytes are terrestrial plants. That means they're not aquatic. They do not grow in the sea water, river water, or anything like that. It grows in the land, right? They are terrestrial. Okay, they're terrestrial and they are epiphytes. What do you mean by epiphytes? Epiphytes are like the hanging plants. Not the hanging plants, actually plants which are attached to another tree or something. For example, if you take orchids are epiphytes. If you take an orchid, we have a tree like this and we grow the orchid attached to another tree, right? That's how the orchids grow, right? So likewise, lycophytes are epiphytes, okay? Orchids are also epiphytes. And lycophytes produce upright stems as well as ground-hugging stems. There are 2 types of stems produced. One type of lycophytes produce the upright stems. Stems are upright like this. And the other type of lycophytes produce the ground-hugging stems. They are going all over the ground, right? In the upright stems, obviously, in the upright stems only leaves are found, right? Small leaves can be found in the upright stems. In the ground stems, can you even believe that, can you even think that the leaves can be found? We trample these areas, insects, animals walk on those areas. I do not think there are leaves as much as the upright stems, but we are not learning about the structural, the structures of those stems and all. Just know that these upright stems contain small leaves, but they haven't told about ground-hugging stems leaves. They have just told us they are just branching, dichotomously branching, like a dichotomous key. You all have heard about the dichotomous key, right? Dichotomous means it has so many branches like this, right? It gets divided into several parts. All right. And also these lycophytes have these structures called strobili. All right, they have these structures called strobili. And remember, lycophytes can either be homosporous or heterosporous. Selaginella is homosporous, right? Selaginella is homosporous, that is a spike moss. Remember, all spike mosses Selaginella is heterosporous. All spike mosses are heterosporous. Do you know how I remembered this? I will tell you how I remembered this. When I saw now, all club mosses are homosporous. That's what they have given in our resource book, right? All club mosses are homosporous. They have given Lycopodium as the example. And they have given all spike mosses are heterosporous. And they have given Selaginella as the example. See, S and S, right? So all spike mosses means Selaginella. That's how I remember. Spike, S and S, Selaginella. And heterosporous, when you pronounce heterosporous, it also has a bit of an S emphasis on that word, right? Just pronounce heterosporous. You pronounce heterosporous. Can you hear a more of a S sound in that? Rather than when you're pronouncing homosporous. When you speak heterosporous, it's like, that's how, by the pronunciation, I use those techniques. Spike mosses are heterosporous. Example, Selaginella. I use those small techniques. I don't know whether it suits you. These are the techniques that suits me. These are simple techniques that I used. I will come to one of the major techniques that I use later. But now here to study all these small parts, I use simple techniques like this. So I know all spike mosses are heterosporous. Example, Selaginella. And the other thing is the opposite then. All club mosses are homosporous. The remaining example is Lycopodium. That's how I remembered it. So I just gave you a revision of the lycophytes. Let's go through the answers. They're asking us the incorrect statement. Children, guys, what I learned from my chemistry teacher, one thing that I learned from my chemistry teacher, it helped me a lot in my exam. You have to underline the questions, especially if they bold the incorrect or the correct part. But make sure, even if it is bold, underline the incorrect, because when you are in that exam condition, you forget the question. Even though it's in bold, sometimes you, as soon as you see the correct answer, you mark it as correct and you come. But at the end, they have asked the incorrect answer. So for that, you have to always underline it and be mindful. Okay. Incorrect statement about phylum Lycophyte. Number 1 is dominant plant is sporophyte. Yes, the dominant plant is sporophyte. It is directly given in your resource book in page number 90 in, I think, unit 4, right? Unit 3 or 4, unit 4, yeah, page number 90, unit 4, they have given dominant plant is sporophyte. Strobili are found in club mosses and for spike mosses. Yeah, there is no problem for that. Sorry, there is a problem because not in all. Now, children, when I was doing my A-levels, I classified these statements into many, most, some, all. In our resource book, there are many statements like this. Some statement starts with many. Some statement starts with more. Some statement starts with some statement starts with all. These are heavily checked in your MCQ paper. In this MCQ paper also, they have checked now. Can you see? I also, without reading the whole answer, since I finished my A-levels, it's okay for me. But for you guys, it's not going to be okay, because you guys are still doing your A-levels and you've got 3 months, so you would be very careful with this. So strobili are found in all club mosses and spike mosses. It might seem as correct. Yeah, we have learned, right? In Lycophyte, strobili is there. We have learned. Yeah, we have learned. You might think like that, but you haven't carefully gone through the book. It's not in all club mosses. In many club mosses and spike mosses, strobili is there. In many, okay, not in all, in many. So while you are reading this resource book, one thing that I did was, I do not know whether I have those classified many, more, some, all, but I'll try to classify it for you guys now. Okay, so from next month onwards, I'll give you those small notes as well, which I use, and also these notes that I make for you guys, especially classifying the many most in your resource book. Unit by unit, I'll try to give. Okay, so in many club mosses and spike mosses, only strobiluae are present, right? So the second one is correct. Second one is incorrect because of one word, just one word. Maybe homosporous. Yeah, lycophytes may be homosporous. What are the homosporous lycophytes? Club mosses. All club mosses are homosporous. All spike mosses are heterosporous. I said small leaves are found in upright stems. That's what I said, right? I drew diagrams to explain you that. I said the ground hugging stems are dichotomously branched, right? That's what I explained it to you. All right, some are epiphytes. Exactly. Now the word some is also important, children, because this can also be an incorrect one. Sometimes some questions have 2-3 answers, right? That's why they give the answers 2 or 3, 3 or 4. If all the answers are incorrect, then they give all, right? So you have to be very careful. Otherwise, you'll get confused, right? Some are epiphytes. Some are epiphytes. That is true. In lycophytes, not all are epiphytes. Only some. That word is there in page number 90. I'll try my best to classify these points into some, many, most, and all, and give it to you by next month. I'll try my best, okay? So the answer is the answer is the second one. Shall we write the review for the second one? Okay, write down the review. All club mosses are homosporous. All club mosses are homosporous. Example, Lycopodium. When you write Lycopodium, L capital, and it should be underlined. Okay, it should be underlined. Lycopodium should be underlined. Selaginella should be underlined, right? And then write the next one. All spike mosses. When you're writing answers for SCQ also, make sure you underline those words, okay? Selaginella, Pogonatum, Cycas. Sorry, yeah. Is my screen shared? Suddenly something happened. Yeah, my screen is shared. Okay, all right. Okay, now I was telling you the review. All club mosses are homosporous. Example, Lycopodium. Second one, all spike mosses are heterosporous. Example, Selaginella. All spike mosses are heterosporous. Example, Selaginella. All spike mosses are heterosporous. Example, Selaginella. And write down this point as well. Some lycophytes are epiphytes. Some lycophytes are epiphytes. Lycophytes means the ones belonging to phylum Lycophyte, okay? Some lycophytes are epiphytes. And you can also write the next point, In many club mosses, highlight the word many. In many club mosses and spike mosses, sporophylls are clustered. In many club mosses and spike mosses, sporophylls are clustered into club-shaped cones called strobiluae. In many club mosses and spike mosses, sporophylls are clustered into club-shaped cones or strobiluae. In many club mosses and spike mosses, sporophylls are clustered into club-shaped cones or strobiluae. Right? I'll repeat it again just in case some of you didn't write it. In many club mosses and spike mosses, sporophylls are clustered into club-shaped cones or strobiluae, right? So that is the review. So we've done the 8th question, and this is one of the questions that I was waiting to discuss with you guys. One of my favorite questions. Now you might think all are my favorite questions. Yes, you should also make these questions your favorite. Phylum Protista. They are asking who introduced the phylum Protista. It is actually Ernest Haeckel. Now there is nothing to discuss in this. But I have something very important to tell you guys. A small technique that I used to study this. Now in your third unit, when you take this, you have all the scientist names with their foundings and all, right? And it's a bit difficult to remember all these things. So I came up with some mnemonics. These are made by myself while I was studying. I made this by myself, and I use these small tactics to remember these small points, because otherwise I can't keep everything in my head for a long time, right? So I use the small techniques. I used the word EPP. You can also use this, the word EPP. Write down now if you like, because I won't give you a review on, yeah, I will give you a review. It's okay. Don't write it now then. If you want, you can write it down. EPP. What do you mean by EPP? E stands for Ernest Haeckel, right? Ernest Haeckel. E stands for Ernest Haeckel. He introduced the kingdom Protista. He introduced the kingdom Protista. And also he introduced the taxon phylum. He introduced the taxon phylum. This is a small shortcut method which I used to study this. It was made by myself. I did not have any other mnemonics to use. That's why I used EPP. E stands for Ernest Haeckel, P stands for Protista, and the other P stands for phylum. That means Ernest Haeckel introduced the kingdom Protista and the taxon phylum, and he classified many organisms into the kingdom Protista as well as into the taxon phylum. Not only this, I want to tell you another thing. The other thing is RK. If you know about SRK, that is how I remembered. RK is an easy word, easy mnemonic to remember, RK, because SRK is there, and then I quickly got it into my head, okay, RK. What does RK stands for? R stands for, now you should be able to guess, Robert H. Whittaker. R stands for Robert H. Whittaker. What did he introduce? He introduced the 5 kingdom. K stands for kingdom. He introduced the 5 kingdom system of biological classifications. So R stands for Robert H. Whittaker. K stands for the 5 kingdom system of biological classification. What are the 5 kingdoms? Monera, Protista, Monera. Those are not the kingdoms now, okay? Monera, Protista, fungi, Plantae, Animalia. So he only made this 5 kingdom system, right? Monera, Protista, fungi, Plantae, or Animalia. Not only this, I have another mnemonic to tell you. The other mnemonic that I wanted to tell was CD. CD is also easy to remember, right? What do you mean by C? C stands for Carl Woese. Carl Woese. What did he introduce? He introduced the 3-domain system. He introduced the 3-domain system. So Carl Woese introduced the 3-domain system. So did you see how easily I studied all these parts? I didn't use anything. I didn't use any hard. I didn't use any hard method to study this. I just used some easy techniques to study all these parts, right? So you should also use these techniques, since I've told you. What are the 3 domains, guys? The 3 domains are domain bacteria, domain archaea, and domain eukarya, right? Domain bacteria, domain archaea, and domain eukarya are the 3 kingdoms. So CD stands for Carl Woese and 3-domain system, and RK stands for Robert H. Whittaker and the 5 kingdom system, and EPP stands for Ernest Haeckel, introducing taxon phylum, and introducing the kingdom Protista, right? So that is a small technique. Shall we write down that technique in the review? Write down capital E, capital P, and write down this. Ernest Haeckel introduced the 3rd kingdom Protista. Ernest Haeckel introduced the 3rd kingdom Protista, and taxon phylum. Ernest Haeckel introduced the third kingdom protista. Write down question number 9 and write this. EPP, Ernest Haeckel introduced the third kingdom protista and taxon phylum. And taxon phylum and classified many organisms. Ernest Haeckel introduced the third kingdom protista and classified and taxon phylum and classified many organisms. Right? Then write capital R, capital K. Write capital R, write capital K, and write down Robert H. Whittaker. Robert H. Whittaker. Robert H. Whittaker introduced the 5 kingdom system of biological classification. Robert H. Whittaker introduced the 5 kingdom system of classification. Robert H. Whittaker introduced the 5 kingdom system of classification. Robert H. Whittaker introduced the 5 kingdom system of classification. Write down the 5 kingdoms. Monera, Protista, Fungi, Plantae, and Animalia. Monera, Protista, Fungi, Plantae, and Animalia. Monera, Protista, Fungi, Plantae, and Animalia. And then write down capital C, capital D, and write down Carl Woese introduced the 3-domain system. Carl Woese introduced the 3-domain system. Carl Woese introduced the 3-domain system. If any one of you wants the recording, you can request me. After the class, I can give you the recording, but I have to make sure that it gets successfully recorded because sometimes I have some technical issues. Due to that, I really hope that I would be able to record it successfully. Okay. All right. And if you want it, if you join the class late, because I explained some important tips in the beginning. So if you join the class late, you can ask for the recording. No worries. Okay. If I successfully record it, I will share it with you. All right. Now we did the 9th question. Now let's go to the 10th one. Select the answers contain characteristics possessed by a single phylum. I will go fast now since it's 10:43. I might have to go a bit fast. Okay. All right. Ectotherm. Now they are asking about some characteristics which are possessed by one phylum. They are talking about now by reading all these things, we can understand they're talking about phylum Chordata. They're talking about phylum Chordata. If you read all these 5 answers, ectothermic, caudal fin, moist skin, swim bladder, nictitating membrane, scales, differentiated teeth, digits, all these are, all these are characteristics possessed by phylum Chordata, right? But actually, it's like this. If you take the A statement, they're asking if the A statement is there in the single phylum. If we take ectothermic and caudal fin is heterocercal. All right. Now, this question, to tell you the truth, this question is a bit confusing. When I was doing the question back in 2023, I got this wrong also. This is a bit confusing. All right. Caudal fin, we know caudal fin is heterocercal in which class? Caudal fin is heterocercal. I'm not talking about the phylum, I'm talking about the class. Caudal fin is heterocercal in which class? They are heterocercal in class chondrich ties, right? Chondrich ties, you know about the ostage ties and the chondrich ties, right? Chondrich ties. Okay, chondrich ties, they're heterocercal in the chondrich ties. And ectothermic. All fish are actually ectothermic. Okay, all fish are actually ectothermic. All fish in the sense chondrich ties, ostage ties, all are ectothermic. That is not in your resource book. I got it from the internet. Okay, I searched the internet and I found that all fish are ectothermic. That is not in your resource book. So this statement seems like correct because they are found in one class, but actually they are asking about a single phylum. Let's keep the question for a question apart because they have given the answer second one. Now let's go to the B part. Moist skin with glands and limbs with digits. Children, in provincial papers, they make many mistakes, right? Because there are no people to double check. I mean, there are people, but they do not take it much seriously. I have seen in many of the provincial papers that have practiced last year, there are many mistakes that they make, especially in typing. All right, so people get confused when doing the paper, but if you find there is an error, you just have to skip it and go, okay? Moist, and that happens in that happened in 2023 level also. There was a wrong question from genetics. As soon as I saw the question and I did it once, I understood, okay, the answer is not here. The answer is not here. I think I got 21 or so. I don't remember the value. I'll just say as an example, if I got 21, answer 20 was there in that genetics question. So I underlined that answer question. I wasn't even worried because from the look itself, I knew it was a wrong question. So by the glance, by having one glance, you should be able to identify whether it's a wrong one or the correct one, right? For that, you have to practice papers. Okay. Now the second one, give me a second. I'll drink some water. You can also have a small water break. I will stop sharing. You can have a water break of 2 minutes. I want to check the participants. All right, now I'm going to share the screen again. I hope you guys can see the screen now. Now let's go to the B part. Moist skin with glands and limbs with digits. Now who has moist skin out from all the animals? Moist skin is there in amphibians, right? Class Amphibia. Amphibians, or the class Amphibia. And limbs with digits. Limbs with digits. Amphibians also, actually these digit words come under reptilia. If you're reading the resource book carefully, the digit word is not found in amphibia. But since they have given us the second one as the answer, I think this should be an all question. But since they have given second one as the answer, we should go with that way, okay? They do have digits, right? What do you mean by digits? We have feet and we have toes and we have fingers, right? They have this stuff called digits, okay? And the third one, having a swim bladder, and we can see both these are in class Amphibia. In A part also, both these features are in class Chondrichthyes, right? If you take the C part, having a swim bladder. A swim bladder. Who has the swim bladder? Class Chondrichthyes have swim bladder, right? They have a swim bladder. Then who have the nictitating membrane? Class Amphibia has the nictitating membrane, right? So the C1 includes two classes, not the same class. This includes one class. These two in the B1 also the characteristics are included in one class, but here they have talked about two classes. Therefore, we can exclude it. We can convert this question into single class actually, not phylum. Single class, because phylum is phylum Chordata. If you want to make the question correct, you can convert it into class, okay? Then only the question makes sense. Then go to the D1, no glands with skin and limbs with digits. Okay, do reptiles have glands? No, right? Who has glands? We, mammals have glands, right? Mammals have glands, and but reptiles do have limbs with digits. But since they do not have glands, we can't take this answer. Then the E1, legs have scales. Again, who have scales, guys? Reptilia, class Reptilia have scales. Who have differentiated teeth? Mammals, right? Class Mammalia are the only class which have differentiated teeth, right? They are the only ones which have differentiated teeth, and that is why they are called heterodonts. That is not there in your resource book. This is a general knowledge one that I'm going to tell you. They are called heterodonts because they are the only ones which contains differentiated teeth. What do you mean by the differentiated teeth? We have incisors, canines, premolars, and molars, right? We have those 4 teeth in our mouth, right? In our buccal cavity, right? umbilical cavity, right? Yeah. Okay. Now, so A, B, and D are the correct answers. Therefore, the correct answer is 2. Shall we write the review quickly? All right, please write the review quickly because we don't have much time. Conrich thighs and Ostage thighs are ectothermic. Conrich thighs and Ostage thighs are ectothermic. All right, then write the second point. Only mammals have differentiated teeth. Only mammals have differentiated teeth. They are called heterodonts. They are called heterodonts. Only mammals have differentiated teeth. They are called heterodonts. Only mammals have differentiated teeth. They are called heterodonts. Only mammals have differentiated teeth. They are called heterodonts. Right, the third point. Scales are present in class Conrich thighs, class Ostage thighs, and class Reptilia. They have said legs have scales, right? Usually scales are there in all the fish as well as Reptilia. So scales are present in class Conrich thighs, class Ostage thighs, and class Reptilia, right? So I hope you wrote the review. I'll quickly repeat it just to make sure you wrote everything. Conrich thighs and Ostage thighs are ectothermic. What do you mean by ectothermic? Children, I didn't explain you that, right? Ectothermic means they can change their body temperature with the surrounding temperature. If the surrounding temperature is hot, they can cool it down, right? We also do that. No, I mean, yeah, they can change the temperature with the body temperature, with the external temperature, right? We can't change the temperature. We do have a process called homeostasis, but it is not what I meant by the word ectothermic. We and also the birds and the not the amphibians. We and the birds are endothermic. Okay, we and the birds are endothermic. Mammals and birds are endothermic. Ectothermic means they can change the body temperature according to the environment, and they can get adapted to that environment. That is why, because fish, when they go to fish can survive in cold regions as well, right? Even if suddenly it gets cold, they can survive in the cold regions. But fish do migrate as well, but somehow up to a certain level, they can control their temperatures, and that is why with the environment, that is why they are called ectothermic. Okay, now let's go to the 11th one. I badly wanted to explain you the reproductive cycle of fungi, but due to limited time, but it's okay, I'll try to explain you. The following structures are found in different phyla. Select an answer which includes examples of each phyla. You have to read the question carefully. You can't read like me. Make sure you read each and every word carefully. That is my advice for you, okay? Mycelium is dikaryotic. Now, children, in our resource book, they have talked about this dikaryotic phase only in phylum Basidiomycota. Only in phylum Basidiomycota have they talked about the word dikaryotic. But to tell you the truth, both ascomites and basidiomycetes have the dikaryotic stage in their cell cycle. So what is agaricus? Agaricus is a Basidiomycota, right? Agaricus is a Basidiomycota. Therefore, A is correct. That one we can take. This part is correct. Let's put a tick. Penicillium. Penicillium is a Ascomycota, right? Ascomycota, it is also correct. Why? Because even though in our resource book they have said, they don't usually ask questions like this. Since this is a provincial paper, they have asked you. But anyhow, it's better to remember this thing in your head, right? Both Ascomycota and Basidiomycota have dikaryotic stage. Okay, other fungi also have dikaryotic stage, but dikaryotic stages are seen especially in Ascomycota and Basidiomycota. Okay, so mycelium, but in our resource book, they have talked about only Basidiomycota. So by mycelium is dikaryotic, agaricus is, since it's a Basidiomycota, I took it as correct. And Penicillium might take it as correct, but Chytridium, it belongs to phylum Chytridiomycota, right? So Chytridiomycota, I didn't tell you that it has dikaryotic mycelium, so we can easily cut the third answer off, right? So say I eliminated it. I didn't even look at the other answer, but you should look at the other answers. I didn't have time, that's why I'm going to go fast now, right? The fourth answer, again, they have said Agaricus. What is Agaricus? It belongs to phylum Basidiomycota, therefore correct. Then what is Mucor? Mucor belongs to phylum Zygomycota, therefore we can easily cut it off. Then produces Asci. Which one produces Asci? Asci is produced by the word itself, we know it's produced by Ascomycota, right? What are the Ascomycota? We know Aspergillus belongs to Ascomycota. Does Rhizopus belong to Ascomycota? No. Rhizopus belongs to what? Phylum Zygomycota, right? Zygomycota. You can easily cut the one as well, and now all the remaining ones are the second and fourth one. Now let's go through the C part. Produce zygosporangium. Which one produces zygosporangium? Is it Basidiomycota? Is it Ascomycota? Is it Zygomycota? Is it Chytridiomycota? It's Zygomycota. What are the examples in Zygomycota? It's Mucor and Rhizopus. So we can easily see the fourth one is the answer. Why? Because Mucor is Zygomycota, but Saccharomyces is actually belonging to Ascomycota, right? And Chytridium are the ones which produces flagellated spores. So if you didn't know that Mucor didn't belong to Zygomycota, you will be stuck in between the second one and the fourth one. That is why you should know all the examples very clearly. This is not a hard question. This is an easy question. Guys, I badly wanted you to show this life cycle to explain you about this dikaryotic stage and all, since most of you all had just memorized the resource book, but you guys don't know even what is dikaryotic stage. While I was doing, I was so interested. Like, you should be interested, okay? Interest only builds up your passion towards biology. When you like biology, you can easily improve your marks. So you have to know what this dikaryotic stage is as well, even though it is not that important. So I badly wanted to explain this to you, but I will send a small example in the group today to explain that, and I will try to give a voice recording as well explaining that. I'm not going to do it now, okay? I will try to give it by the end of the day today. All right, now let's go to the 12th part. Characteristics of different phyla of seed plants are given below. What pair is correct? We didn't write the review, right? For the 11th question, write down the review. Write down the review. Dikaryotic stages are seen. Dikaryotic stages are seen, especially in ascomycetes, or you can write phylum Ascomycota. Dikaryotic stages are seen especially in phylum Ascomycota and phylum Basidiomycota. Okay, my screen sharing has ended. I'm really sorry, I couldn't. Can anyone tell me from where did you stop? From where did the screen sharing stop? So that I can repeat it quickly. I'm really sorry. Extremely sorry. Can someone please tell me? So that I can explain that part again. But I hope that you could hear my voice all this time, right? Okay, that means you didn't hear the MCQ 11 at all. Okay, guys, I didn't draw anything in, I didn't draw anything in the question number 11. Thank you very much for replying. I'm very grateful to that. Thank you very much. And now I will start from question number 11 again. I will quickly go through that. Guys, mycelium is dikaryotic in either phylum Ascomycota or in phylum Basidiomycota. Okay, they are dikaryotic in either Ascomycota or Basidiomycota. But in your resource book, they have only talked about dikaryotic with the Basidiomycota. But for your general knowledge, I'm going to tell you, knowing these two things are important, right? So mycelium is dikaryotic. We know mycelium is dikaryotic in Ascomycota and Basidiomycota. Now let's go through the answers. Agaricus, Penicillium. Let's go through the first answers. The A part at first. Agaricus, Penicillium, Chytridium, Agaricus, Mucor. What is Agaricus? Agaricus belongs to phylum Basidiomycota. Agaricus belongs to phylum Basidiomycota, right? So you can easily tick that off. Agaricus belongs to phylum Basidiomycota. That means it has a dikaryotic stage. But this Chytridium and Mucor, but to which phylum this Chytridium belongs to? Chytridium belongs to Chytridiomycota. Mucus belongs to what? Mucor belongs to phylum Zygomycota, right? Zygomycota. Then Penicillium belongs to Ascomycota, right? So therefore, 1, 2, 4 are correct. You can easily cut the third and fifth one off. Did you understand why I cut 3 and 5? Because from the A part itself, I understood that the third and fifth was wrong. But you guys, when you're doing the paper, check the other answers also. Because since I'm in a rush only, I'm trying to finish the paper. That's why I'm going a bit fast. I'm not going to look at the other answers, but you guys should look at the other answers, okay? Then only you can be happy with your answer. You can face the other question in a happy mood, in a good mood, only if you confirm that your answer is 100% correct. Now, let's go to the B part. B part says, now we know it's 1, 2, or 4, right? 1, 2, or 4. They say produce ASCII. What produces ASCII? Ascomycota. What are the Ascomycota? Aspergillus is Ascomycota, right? Rhizopus is not Ascomycota. Rhizopus is what? It is Zygomycota. Penicillium is not. Yeah, we don't have penicillium. We have already cut the fourth one off. So we do not have to worry about that. Third and fifth one are cut off. So we do not have to worry about that, right? And now we can easily cut one also because Rhizopus belongs to phylum Zygomycota. Now let's go to the C part. Produces Zygosporangium. What produces Zygosporangium? Phylum Zygomycota produces Zygosporangium. What are the two examples that they have given on the Zygosporangium? Mucor and Rhizopus. You can easily see that the answer is the fourth one because Saccharomyces. Sorry, here also they are no. Wait, here also they have given mucus, so we still can't find the correct answer. We have to go to the final answer to check what is the answer. Produce flagellated zoospores. There is only one phylum which produces flagellated zoospores, and that is what? Flagellated zoospores are produced only in phylum Chytridiomycota. Therefore, the answer is Chytridium. So the answer is the fourth one, right? I will give you, that's all I explained. I just drew these, annotations. That's it. I wanted to explain you the dikaryotic stage, but I don't have time for that. So I will give you a voice recording with a small image today. Okay? All right, write down the review. Dikaryotic stages are seen. Dikaryotic stages are seen. Dikaryotic stages are seen, especially in Ascomycota and Basidiomycota. Dikaryotic stages are seen, especially in phylum Ascomycota and Basidiomycota. Dikaryotic stages are seen in phylum Ascomycota and Basidiomycota. That's all you have to write. I had a big review to give actually. I wanted to give you the examples of Basidio, Asco, Zygo, Chytridio, but no time. You have to write your own review. I'll tell you how to do that at the end. 12th one. Characteristics of different phyla of seed plants are given below. Okay, guys. Will you mind if I carry the session until 12 o'clock? Can I have an answer from all of you? Does anyone wants to leave the session? Does anyone wants to leave the session? Is it okay if I carry the session until 12 o'clock? I'm taking 3 hours, that means. If you have any... Okay, I will carry... Okay, one person has replied. Thank you very much. Are the others okay with me carrying it until 12 o'clock? Okay, thank you very much. All right. Now, I will go through the 12th one, characteristics of different phyla of seed plants. This is a very easy topic because we know there's no much that means they haven't given much theory in this area, right? Neophyta, Cycadophyta, Coniferophyta, Anthophyta. These things are very easy. They have given us with beautiful pictures also in our resource book. Not beautiful pictures, but yeah, it's good. We can get an idea, right? Then what is this phylum? Neophyta. Remember guys, phylum Neophyta They are the only gymnosperm which have vessels in the xylem. That means they are the only gymnosperms that have xylem vessels. Other gymnosperms have only tracheids. Okay, other gymnosperms have only tracheids. Only angiosperms and Neophyta have xylem vessels. If you want, you can write it now itself. Write the review now while I'm doing the question. Only Neophyta and angiosperms. Only Neophyta and angiosperms have xylem vessels. Only Neophyta and angiosperms have xylem vessels. Only Neophyta and angiosperms have xylem vessels. And also in gymnosperms, guys, the leaves actually look like those of flowering plants. You know the normal flowering plants leaves, right? They look like the leaf. The leaves look like just like the leaves in the flowering plants, and they're seeds, right? They have these seeds, right? When we see these seeds, we think they are fruits. They are just seeds, but as soon as we see the seed, we think they are fruits. Why? Because they look like fruits of angiosperms. So you can see A1, leaves look like those of flowering parts. That is true. What is this Cycadophyta? Cycadophyta is actually dioecious. Do you know what the meaning of dioecious? Dioecious means unisexual. What do you mean by unisexual? Separate male plant, separate female plant. And that is cycas, right? I call it cycas. You can call it cycas if you want. All right, I call it cycas. So they have a separate male plant and they have a separate female plant. That's why they're called dioecious, or dioecious, another word is unisexual. Unisexual. This is the same word. Then monoecious means bisexual. Bisexual means having both male and female parts in the same plant. Here, there are separate male and female plants. Cycadophyta is unisexual, and they are the ones which have flagellated sperms. If you go through your resource book, you can identify that statement. They have flagellated sperms similar to seedless vascular plants. Now here we are talking about the seed plants, right? That's why we call it gymnosperms, right? Seed plants, we're talking about the gymnosperms. But actually, they have flagellated sperms just like the seedless plants. Okay. So number 2 is also, B is also correct. Now let's go to Coniferophyta. Coniferophyta, now I told you Cycas is actually unisexual, but Coniferophyta is bisexual. In the same plant, two types of spores are produced in separate cones. That means two separate cones are produced, male cone and female cone. In Coniferophyta, a male cone is produced in the same plant, female cone is produced in the same plant. Female cone produces female spores, male cone produces male spores. So two types of spores are produced by one cone, it's wrong. Two types of spores are produced by two cones because there are separate male cones and separate female cones in the same plant. And that is why we call it bisexual or monoecious. I hope you understood the difference. So third one is wrong. Anthophyta, they have persistent floral parts. What do you mean by persistent? In English, persistent means it usually lasts for a long time, right? And also if you go through the phylum Anthophyta or the angiosperms, in that part, they have said a fruit. How is a fruit formed? After the ovary is fertilized only, a fruit is formed, right? So a fruit typically consists of a fertilized ovary and it consists other persistent floral parts. For example, if you take the fruit, you can sometimes see the sepal, right? You can sometimes see the sepal in that fruit. If you take a tomato, you can see the sepal, right? Those are the persistent floral parts. And we are learning about the persistent floral parts under angiosperms. When we are learning that Anthophyta reproductive structure, we are learning what those floral parts are: stamens, petals, carpels, and also sepals, right? We are learning about those things. We have learned about those things when we did our theory. I mean, you guys might have learned about those things. So yeah, they have persistent floral parts, and that is correct. Neophyta, seeds looks like seeds of angiosperms. No, no, no. When we see a neophyta seed, we think it's a fruit. So they don't look like seeds. They look like fruits of angiosperms, right? So that is basically the 12th question. I will give you a small review on that question, a very small review. Write down Neophyta. Neophyta is the only gymnosperm. Neophyta is the only gymnosperm that have vessels in the xylem. Neophyta is the only gymnosperm that have vessels in the xylem. Neophyta is the only gymnosperm that have vessels in the xylem. All right. That is enough for now. Let me check whether the screen has been shared. Okay. Now let's go to the 13th one. Which statement is correct about the xylem tissue? This xylem tissue is a very interesting part that we have got. Let's read one by one and understand. Perforation plates are present in the vertical walls of the xylem elements. Children, it's like this actually. Now the vessel elements are like this. These are the perforation plates. Vessel element combined together to form the xylem tube like this. These are separate vessel elements. We don't call these cells. Why? Because they're not living. We call these vessel elements. These vessel elements forms the xylem vessel, and on under each and every vessel element there is a perforation plate, right? So they are not vertical. Vertical means this side, right? Vertical means the upright side. Horizontal means this side. They are horizontal. So that is wrong. The first statement is wrong. Perforation plates are not present in the vertical walls. They are present in the horizontal walls, right? So that is wrong. Tracheids are long, thin cells with tapering ends. Yes, we know that xylem tissue has 4 types of cells. They have what? Xylem vessel elements. They have tracheids. They have sclerenchyma cells, and they have parenchyma cells as well, right? These are the 4. These are the 4 cells in the xylem, right? We have learned about those things in our theory in Unit 4. Right, and then there are long, thin cells with tapering ends. Tracheids are long, thin cells with tapering ends. That is true. But vessel elements also, they have said they are cylindrical and long. Are they longer than tracheids or are they shorter than tracheids? Remember, if they give you vessel elements, if they give an MCQ like this, vessel elements are cylindrical and long. That is true. But remember, they are wider and shorter than the tracheids. They are shorter than the tracheids. Even though they are long, they are not as long as tracheids. They are wider and they are shorter than tracheids. Okay, that is another. That this is sort of revising. I just don't want to discuss the only the question. I want to go out of the question and discuss the other areas as well, because it should be like a revision for you guys, right? Okay, so B1 is correct. Radial transportation occurs in parenchyma cells. What do you mean by this radial transportation? Radial transportation, guys, it is a transportation from the roots through the cortex into the xylem vessel. Through the roots into the through the roots into the cortex and then into the xylem vessel, right? That is what you call radial transportation. So radial transportation happens through the cortex. Remember, guys, this cortex is mainly made out of, or mostly made out of parenchyma cells. I told you I will give this mostly, many, or some, right? And this is another statement which should be included in that, because cortex is made up of mostly parenchyma cells with intercellular spaces. Therefore, C1 is also correct. Let's go for the D1. Secondary walls of vessel elements and tracheids are thickened by lignin. That is, of course, true. Both tracheids and vessel elements have lignin. Their secondary walls have lignin. That is true. And then the E part, water moves from one cell to another through pits. Actually, in vessel elements, it moves through the perforation plates from one cell to another cell. But in tracheids, they don't have perforation plates. Xylem vessels also have pits, and also tracheids also have pits. In trachea to end through pits. So therefore, we can take E1 also correct. Therefore, the answer is B, C, D, E. That is how you get the answer for such a question. Shall we quickly write down the review? Write down the review. Perforation plates are present. Perforation plates are present at end walls of vessel elements. Perforation plates are present at the end walls of vessel elements. Other walls are interrupted by pits. Perforation plates are present at the end walls of vessel elements. Other walls are interrupted by pits. All right, okay. And if I say if both tracheids and vessel elements have pits, is it true, is it false? It is true. Both tracheids and vessel elements have pits. Now let's go to the 14th question. Incorrect statement regarding the primary structure of the dicotyledonous plant root. Okay, this is under the primary growth of the plant root and stems. Cortex. In the 13th question, I told you cortex is mostly made out of parenchyma cells. That is true. It is given. If I tell you the page number, it is given in page number 115 in unit 4. Page number 115 in unit 4. They have given this exact point. It is made out of parenchyma cells with intercellular spaces. Okay, and also if I draw the structure of the root, it's like this. There is the endodermis, pericycle. You can see vascular tissue is arranged as a solid core. This is core, and since this is solid, we call it a solid core. See? But this is in the middle, that is why we call it core, and this is solid, so we call it, it's not actually solid, but it's made out of lignin and everything, right? The vascular tissues, so it's solid, so vascular tissue is arranged as a solid core. That is true. They're asking us for the incorrect statement. Endodermis consists of a single cell layer. Guys, epidermis is also a single cell layer. Endodermis is also a single cell layer. Both epidermis and endodermis are single cell layer, right? So innermost single cell layer of the cortex is the endodermis. Endodermis consists of suberin, a suberin belt called the Casparian strip, and it has no intercellular spaces in it, right? So those are some extra stuff that you should know. They are in your resource book. So endodermis consists of the single cell layer. It is correct. Pericycle is located outer to the endodermis, which consists of 2 or 3 cells. No. Pericycle is not outside. Pericycle is inner to the endodermis. After the endodermis only, there are 2-3 layers of pericycle. So this statement is wrong. See, just by one word, as soon as we see this, we think it's correct. But this is wrong. Why? Because pericycle is inner to the endodermis. In this paper, most of the answers were made wrong by just one word. They have used only one word to make the answer wrong. So you should be careful. The xylem can be seen as a star shape. Yes, of course. You can see the xylem is in the form of a star, right? Xylem is in the form of the star. This is the phloem, right? So write down the review for that part. Answer is 4th one. I'm going a bit fast now. You can write down cortex is mostly made out of parenchyma cells. Cortex is mostly made out of parenchyma cells. Cortex is mostly made out of parenchyma cells. Cortex mainly stores carbohydrates and transports water and minerals towards the endodermis. Cortex is mostly made out of parenchyma cells. Cortex mainly stores carbohydrates and also transports water and minerals towards the endodermis. Cortex mainly stores carbohydrates and transports water and minerals towards the endodermis. Right. This question is a must question. Even don't go to your, this is what my teacher told me, don't go to your exam without studying this part. This table will come in almost most of the papers. If you take 10 papers, in 9 out from those 10 papers, 9 papers will have this question. A question from this part, from the auxins, gibberellins, cytokinins, abscisic acid, and ethylene. And I will tell you a small way that I used to study this. I think I got the table over here. This is a very beautiful table. It takes time for me to explain this, but I think it works a lot for you, okay? So this is how I studied it. Now, auxin is there, right? You can, okay, before going for auxin, I'll go for gibberellins. Take gibberellins. Gibberellins all starts from stimulate, right? So it's easy. Stimulate stem elongation, stimulate pollen development, stimulate pollen tube growth, stimulate fruit growth, stimulate seed development. And the only one which is not with stimulate is the one which I had to put some extra force and remember. Regulate sex determination and transition from juvenile to adult phase. That belongs to gibberellins. All the other stimulate ones comes to what? Comes to gibberellin, right? But there is a small issue. In other parts also, there is the word stimulate at some point. Stimulate seed germination is over here. But it's okay. Gibberellins also stimulate seed germination. So we know gibberellin stimulates seed germination. So it's okay even if it's here, even if it's there. But here this is stimulate stem elongation in low concentration. Here also it is there. But auxin stimulates stem elongation in low concentration. So when you study the stimulate parts separately and learn that stimulating seed germination is also a part of cytokines. Okay, then they will ask you sometimes they can ask you what are the 2 hormones that stimulate stem elongation. You have studied it. It's in your head. Easily you can recall that. Gibberellins and cytokinins are the ones which stimulate seed germination. And they will ask you another question in another paper like this. What are the hormones which stimulate stem elongation? They won't specify whether it's low concentration or high concentration. They would just ask you that. Then definitely auxin and gibberellins, right? So that is how you should study. That is one way that I study, especially the gibberellins part, okay? And also, I studied in that way. And then comes C1. C1, there is no specific way. You have to study everything over there. But abscisic acid, can you see? Everything starts with promote over here. Here, ethylene is a special one. Even though it has promotes, you can see there are some specific and some special functions are there. Ethylene, it promotes the ripening of many types of fruit. Whenever ethylene is there, what came into my head was pineapple. I don't know why. Pineapple came into my head. So as soon as I see the word fruit somewhere, ripening of many fruits, ethylene. I know that. Promotes flowering in the pineapple family, I know that. That's how I learned those parts. So use techniques to study these things. Don't just memorize these things. If you memorize it, and also guys, one thing, no matter how much you study, this won't stay in your long-term memory. If you can do that, then it's really good. You're really smart, you're really talented. But it's with all the other work, it's really hard to keep. At least once a week you should revise, but it's not possible to revise everything once a week when you got so many things to revise, right? So my advice is, before you go for your model papers, or I'm starting my paper class from next month onwards, if you join to my paper class, before every paper, please study this table and come. So that means every week you're recalling this part again and again, so it goes into your head. And another thing which I did was this senescence part. See, senescence and also I think abscission. Abscission. Senescence and abscission part, I learned it by writing it down. Give me a second. I think I have that. I will show you a picture of my books, okay? My books are actually torn. They are not even in proper conditions, because that much I have used them. Okay, this is what do you mean by leaf senescence? Senescence means, guys, it is deterioration with age. That means it gets old with age. With aging, it gets old, right? It gets damaged. It gets deteriorated. That is what you call senescence. And what do you mean by abscission? You know, abscission means removing something, right? So it is actually the natural. Now, when leaves fall from the tree, that is called abscission. It is abscission. The meaning is natural detachment of parts of the plant. Abscission, the meaning is natural detachment of parts of the plant. When you're studying, you have to go through the meanings also. I will send you a photo of my book into the group. I have written so many things in that, like ways to study and everything. You should also do that. I highlighted the leaf senescence part in a different color. So I know these are the things which are related to leaf senescence. Cytokinins delay leaf senescence. Ethylene enhances and abscisic acid enhances. Then what are the two types of, hormones which promotes leaf senescence or which increases leaf senescence? That is abscisic acid and ethylene, right? So that's how I took the answers. So knowing these things is very important. I think I don't have to give a review for that. It's just you have to remember those things, okay? All right. So the answer for this is 2 or 5. They're asking the incorrect pair. Stimulate pollen development. It's not done by what? It's not done by gibberellins. Pollen. Oh no, stimulate pollen development is done by gibberellin. I think they have made a mistake in it. I took these answers from the Sabaragamu provincial paper. Answer is actually, I think, 5. Answer is actually 5. Regulates development of fruits. What regulates the development of fruits? I think it's okay. Regulates development of fruits is auxin. Actually, this auxin part, guys, I had a big passion towards the auxin part. I always studied it. I really like to study the auxin part because see, functions in phototrophism, functions in gravitropism. When you pronounce these things, you feel a bit of a pulse in you, like you feel like you like it. So you have to have a big passion when you're studying these things. Okay, so the answer is the 5th one. I won't give you a review since it's a table. And for the 16th also, I won't give you a review because 16th question also, you have to take it directly from the book. Death of terminal buds. What is responsible for the death of terminal buds? Okay. Actually, yeah. Calcium is the one which is responsible for the death of terminal buds. All these things are in your resource book. These things should be memorized, but there is a specific way to memorize these things. And also, you need to memorize it constantly. If you memorize it this week, next week also, you have to memorize it. And then after another 2 weeks, you have to make sure you remember it. Again, after another month, you have to make sure you remember it. Again, after 3 months, you have to make sure you remember it. These are some techniques. You can use this. These are psychological techniques, actually. Use those things. So these are direct questions. That's why I'm brushing through these questions. Reduced internode length. I hope my screen is shared. Yes, my screen is shared. Reduced internode length is actually by zinc. Death of leaf tips is by nickel. But I've got to tell you something important over here in this table. Let me take the table. When it comes to molybdenum, nickel and molybdenum and nickel, the deficiency symptom was death of root tips and shoot tips. Chlorosis in all the leaves. In nickel, death of leaf tips. Death of leaf tips, chlorosis in all the leaves. How did I remember this? Molybdenum is death of root and shoot tips. See, root has an O. Root has double O. Shoot has double O. Molybdenum has an O. So molybdenum, death of root and shoot tips, chlorosis in all the leaves. Nickel, death of leaf tips. That's how I remembered it. See, I have used small tricks. So you guys can also use these tricks to keep everything in your mind, right? So the answer is the second one for the 16th question. While I'm discussing the next few papers also next time, I will go through these things. If I cannot finish the paper discussion today, I will try to send a recording of the full paper discussion. I will try this today. What happened was actually I want to discuss everything with you. Out from the question also, I wanted to go to revise this part since this is the very first day. But from next day onwards, I will adhere only to the question, and I will only teach the question part. And I will give you small reviews because the the point of giving review is because I want to keep you in the track during the class hours. After the class hours, I will tell you how to write a specific review. Right, you have to do your reviewing by yourself. I will tell you how to do that. Today I will tell you. Think if I cannot finish the class, we have 30 more questions. If I cannot finish the class, I will send you a recording of the full paper. All right. Okay. The following are some characteristics of life cycles of 3 plant species. Gametophytes are monoecious. Okay. Gametophytes are dioecious in Pogonatum. What do you mean by dioecious? Dioecious means unisexual. Unisexual means what? Unisexual means separate male, separate female plants. There is a female and male gametophyte. But gametophytes are monoecious in Nephrolepis. So you can cut out the Pogonatum answer. You can keep the Nephrolepis part. And the remaining ones are with Cycas, right? Cycas gametophyte is dioecious. Again, it is unisexual. So you can easily cut off the third and fourth one as well. I want you guys to take a screenshot of this part, because this part is important. If you can, go home and write down the review as this part. This is the review that you need to write. This is something that I made by myself by revising, by reading all the pages in the resource book. So you can take a screenshot of this part and you can write this later and you can study this. All the, like, you know, which parts are the which parts are the dioecious one, which parts are the monoecious one. I have clearly said in this description, right? So third, fourth, fifth answers are gone now. And also we know sporophytes have fiddlehead young leaves in Nephrolepis, right? Then female gametophytes are photosynthetic. Sporophyte is dominant, herbaceous, and have vascular tissue. Female gametophytes are photosynthetic. That we can understand. It is from Selaginella, because Pogonatum we didn't learn anything about, you know, we didn't learn that sporophytes are dominant, right? In Pogonatum, give me a second. I have to charge my tab. All right, so I was at the second part. Herbaceous, and we know gametophyte is dominant in Pogonatum. So you can easily cut off the first one. See, even if you didn't know much of a theory, I know these theories are very confusing because you have a lot of theory points in that, right? But even if you didn't know the theory points very well, you can answer this by specific points, you know, right? Sporophytes are dominant, not Pogonatum, it's the gametophyte which is dominant. Sporophytes are dominant in Nephrolepis, Selaginella, and Cycas, right? Herbaceous and have vascular tissue, then definitely Selaginella. Then they are saying gametophytes are dominant. What is the only plant where we learn that gametophytes are dominant? It's only in Pogonatum. Therefore, you can easily select the second one as the correct answer, right? And write down this review. Dioecious means, do I have this dioecious? It's okay, right? Dioecious means, write down this review at home, please. Dioecious means unisexual. Monoecious means bisexual. Right. Dioecious means unisexual. Monoecious means bisexual. Now they're asking not a plant defense mechanism from pests and pathogens. Okay, so they're basically talking about the biotic stresses, right? Biotic stresses. Okay, so formation of cork. We know if, like, you know, the reason after an insect or after a pathogen enters a cell, it forms a cork layer. So that in the secondary growth also it forms a cork layer, but these are actually induced structural and chemical defense mechanism. Induced means when a pathogen enters, the cell understands that a pathogen enters. So what happens is a cork layer is formed. So that is true. Then formation of abscission layers. Yes, that is also given in your resource book. It is given as 1 point actually. Formation of cork and formation of abscission layers. They are plant defense mechanism for pests and pathogens. They are actually induced structural and chemical defense mechanisms. That means it wasn't already there. After the insect or the pathogens attack only, it has formed. Okay. All right. Now the third one, morphological changes in the cell wall. Yes, the cell wall changes its structure. Right? The cell wall changes its structure, and that is why morphology means the structural changes. Okay. Morphological changes in the cell wall. So third one is also correct. Develop salt gland. What is this? No, it won't develop salt gland. In salt stress, only salt glands are produced, right? Why do what you call salt glands are developed when a pest or a pathogen attacks? No, that is not correct. From the look itself, from the glance, one glance is enough to understand that the fourth one is the answer. Producing secondary metabolites. Producing secondary metabolites is actually a pre-existing structural and chemical defense mechanism, right? So fourth one is the answer. I think I might have arranged a review for that. Yeah, I haven't arranged a review, but it's okay. Let's start write down a review. Biotic stress. Biotic stress is how plants defend themselves. Biotic stress is how plants defend themselves. Biotic stress is how plants defend itself against pest and pathogen attack. Against pest and pathogen attack. Biotic stress is how plants defend themselves against pest and pathogen attacks. Right. Okay. All right. Now let's quickly go to this is the start of Unit 5. This is where Unit 5 actually starts from, right? The epithelial tissues. Okay. Human epithelial tissues are given below. Simple squamous epithelium. I will send you this photograph also. I wanted to, now this is a short note which I used back when I was studying. This is the short note that I made. I know it's very ugly handwriting, but this short note helped me a lot to understand the differences. I didn't write the same point, but I wrote it in my own words so that I can understand the differences. And then later when I read the points, I memorize them because I have an idea. But without having an idea, if you memorize the points, nothing will go into your head. You can't answer the questions logically. You will be a robot, right? There are many people who answers questions like a robot after memorizing things. Then the problem is when they get a logical question, they won't be able to answer. So that's why I made a table like this. I will send you a photo of this table too, so that you can use to study that. There is nothing I need to explain over here. I just wanted to show you the table, like to show you how I studied and all, right? Okay. Human epithelial tissue and their locations are given below. Simple squamous epithelium. We know simple squamous is a leaky tissue. What you call these gases, sorry, these fluids can easily go through the gases and everything can go through the squamous epithelium very easily, right? Because we know it's a very leaky tissue. It's a single layer and also it's thin and leaky. And we know in blood capillaries, we want this leaky tissue so that the glucose and other substances can move in and out into the interstitial fluid, right? And also alveoli for the gaseous exchange, they need the simple squamous epithelium. Therefore, first one is correct. Then what is this simple cuboidal epithelium? Simple cuboidal epithelium is a bit bigger than the simple squamous, and it has single cuboid, means cuboid is a structure like this, right? Cuboid, oh, cuboid, yeah. No, cuboid is a structure like this, right? No, cuboid is a, wait, now I can't remember the structure. Okay. Anyhow, cuboid means I think it's a structure like this. Cube is a structure. Cuboid is this. This is cube. This is a cuboid. Rectangular 3D structure is the cuboid. Square 3D structure is the cube. Okay. All right. Now, simple cuboidal epithelium. It has dice-shaped cells. Dice-shaped cells, and it is specialized for secretion. Secretions means what? Usually secretory substances are in glands and tubules, right? Glands and tubules. So salivary gland is an example where simple cuboidal epithelium is. Thyroid gland is another example. Kidney tubule is another example. Therefore, answer is correct. Simple column epithelium. Now they are brick-shaped cells, all right, and they are used for selective or active absorption. Where this active absorption takes place, secretion or active absorption mainly takes place in our intestinal lining, right? Therefore, thyroid gland is wrong because thyroid gland has the simple cuboidal epithelium. Therefore, the third one is the wrong answer. Fourth one, pseudostratified. Definitely we know it is found in the nasal passages where cilia is present, right? Trachea, nasal passages, cilia is present. And stratified is the one which usually undergo a lot of abrasion, right? They undergo a lot of, they are the substances that are subjected to abrasion mostly. That means their skin is being peeled off mostly, such as the lining of the mouth, the anus, the vagina, the outer skin, right? So these are correct ones. So the answer is the third one. Write down the review. Simple columnar epithelium is found in the intestinal lining. Simple columnar epithelium is found in the intestinal lining. Simple columnar epithelium is found in the intestinal lining. Okay. Now we'll go to the 20th question. What is incorrect about the human liver? Now, this is the structure of the liver. This is the structure of the liver. The liver is, see, now I'm going to, this is the passage in your resource book. The liver contains 4 lobes. The liver is divided into 4 lobes like this, okay? Each lobe is made out of tiny hexagonal shaped lobules. Can you see? These are in the hexagonal shape. All these lobes are made out of tiny hexagonal shaped lobules, right? And these lobules are the functional unit. These lobules are the functional unit. So if anyone asks you what are the functional unit, the tiny hexagonal shaped lobules are the functional unit. Sometimes some people say hepatocytes are the functional unit. That is false. These the tiny hexagonal shaped parts, this one hexagonal shaped part is the functional unit, right? So, and I didn't read the first part. Its upper and anterior surfaces are smooth. That means this part is smooth, but this part is irregular in outline. All right, that part is irregular in outline. So the posterior, upper, anterior means the up part, posterior means the down part. You don't have to know what you call deeply about these words. These are actually related to body anatomy and everything. So the posterior surface is irregular in outline, having tiny hexagonal shaped lobes. Can you see? With one word, they're trying to confuse us. It's actually made out of tiny hexagonal shaped lobules, not lobes. Therefore, the second one is wrong. Hepatic macrophages are found in the linings of the sinusoids. What are the sinusoids? Now see, hepatocytes are the cuboidal structures like this, small cuboidal structures. They are arranged in pairs. This is one pair, this is one pair. In between 2 pairs, now this is one pair, this is one pair, right? In between 2 pairs is the sinusoid. Right? In between 2 pairs are the sinusoid. They are blood vessels with incomplete, they are actually, sinusoids are blood vessels with incomplete walls containing mixture of blood. They contain mixture of blood. And don't confuse sinusoid with canalicule, I can't pronounce it properly. Canaliculi, bile canaliculi. Don't confuse it with bile canaliculi. Bile canaliculi is found in between two hepatocytes, 2 columns of hepatocytes, not 2 pairs. There are pairs like this. This is one pair, this is one pair. In between a pair, in between two pairs, only sinusoids are formed. In between two columns, bile canaliculi is there, right? If you want, you can draw this diagram in the review. Okay, wait. Okay, you can draw this diagram. If I erase this, the whole diagram is going to get erased. So I'm only going to erase this part. You can draw these two parts to identify the structure of the liver. I'll keep it for 1 minute over here. As the review, you can write it, okay? You can draw it, okay? Then the third one, they say, hepatic macrophages are found in the lining of the sinusoids. Actually, we don't, after we found out that the second one is the wrong answer, we do not have to go through. But since I'm discussing it with you, I'm going through. Even you guys should also go through. Because you are the ones who are doing the paper. To be confirmed that your answer is correct, you have to go through, okay? Hepatic macrophages are found in the lining of the sinusoids. Yeah, see, the statement is there itself. Hepatic macrophages are found in the lining of sinusoids. What are these hepatic macrophages? They're also known as the Kupffer cells. Okay, they're also known as the Kupffer cells. Hepatocytes are arranged in pairs of columns radiating from a central vein. This is the central vein. We can see hepatophytes are arranged as columns like this, right? One pair, another pair, right? Okay, then bile canaliculi run between columns of liver cells. What are these liver cells? Hepatocytes are the liver cells. Yeah, bile canaliculi runs between columns. So that is also true. Answer is the second one. Okay, let's go to the 21st question. Select the correct statement regarding gastritis condition. Glands of the stomach wall are stimulated and secrete excess HCl. Yes, that is true. If you take now, this gastritis question is a question where they do not touch in most of the papers that they give us. I saw this also in this paper during the provincial papers that I did. Mostly this gastritis question doesn't come. I don't know why, but they don't give the gastritis question mostly. But because of that, you have to be aware of that question. That means they can ask you in your paper, in 2024 paper. So, yeah, 2024 paper. So you have to be a bit aware of this gastritis part. So number one is correct. When you secrete excess HCl only, the stomach wall gets damaged because the lining, the mucus layer gets damaged. Okay, so damage to the mucus is also correct. Helicobacter pylori bacteria can be damage to the, not to the mucus, damage to the mucosa actually. Damage to the mucosa layer. All right. Because it says mucus, we can't take that answer. That is why they have given the third one as the answer. See, you have to be careful with specific words. Damage to the mucosa, lining, damage to the mucosa, not to the mucus. Because it's the mucus lining, right? If you damage the mucus, nothing would happen. You should damage the mucus lining. So that lining is called the mucosa. Helicobacter pylori can be infected with long-lasting gastritis. Yes, long-lasting gastritis conditions is associated with the acid tolerance bacteria called Helicobacter pylori. Okay, actually, I was so interested in this gastritis condition because I had gastritis while I was doing my A-levels. I was even admitted to the hospital, so I was so involved in this gastritis part, and I learned that I also have the Helicobacter pylori. So you can easily get it into your head. Okay, now this Saki also said that she has Helicobacter pylori, so you can easily remember it. By life experiences, you can include things into your A-level journey, right? Okay, drugs like aspirin can induce gastritis conditions. Oh, yeah, drugs like aspirin can induce gastritis condition, and also prolonged starvation. If you have starved for a long time without eating, then definitely you can get gastritis. Prolonged starvation is not the reason for gastritis. That is false, right? Therefore, the answer is A, C, and D. Answer is the third one. Write down the review. Longer lasting gastritis conditions. lasting gastritis conditions. Longer lasting gastritis conditions is usually associated with longer lasting gastritis conditions. Longer lasting gastritis conditions is usually associated with longer lasting gastritic conditions is usually associated with longer lasting gastritis condition is usually associated with the infection by the acid tolerance bacterium Helicobacter pylori. When you write the word Helicobacter pylori, H capital, P simple, underline, and don't underline it with one single line. Break the line into two parts and underline it. Helicobacter with one separate line, pylori with another separate line. That is the standard form of writing the words, scientific words, right? Binomial nomenclature. Helicobacter pylori, right? Okay, so the answer is the third one. 22nd one, what is incorrect about the conducting system of the heart? Now, to show that I have shown you the conducting system, and there is a beautiful way to study this part also. Can you see? Now, this was a major guessing in our 2023 A-levels. So we all studied this part. No one studied the diseases like angina, heart attack, stroke. That part wasn't memorized. And no one studied the rhesus system or anything like that. But what I did, no one in the sense, people who went for the guessings did not study that. People who got 3 years definitely studied these parts, right? That's why they could get an A for biology. Bio paper, actually chemistry paper was a bit hard last year. Bio paper was also a bit difficult. Many people said that. So rhesus and angina part, they didn't study. That's why I'm telling you, study everything and go. Use techniques to study these things. One technique that I used to study was SA node is a small mass of specialized cells. Here also see it has the same sentence. First they are saying what is SA node. First they are saying what is AV node. First they are saying what is AV bundle. Then they are telling us the location. Then you should know the location. See, it lies in the myocardium of the right atrium. It lies over here, near the opening of the superior vena cava, right? Or vena cava. I call it vena cava. AV node, it is situated in the wall of the atrial septum. See, AV node, it is situated in the wall of the wall of the atrial septum, and in between the left and right atria. So you can see first they're saying what is SA node, then they're talking about the location, then they're talking about the function, right? That is how you should study this. You should break down into groups. Okay, I know the location. I know that both, I know the, I know the structure of them. They are, both of them are small mass of specialized cells. Then I also know SA node, I also know the location of the SA node. It lies in the myocardium, right? In the right atrium and closer to the superior vena cava, right? And I also know the location of the AV node. It is situated in the wall of the atrial septum. You have to question yourself. You have to ask questions and study these parts. That's how you get it into your head, okay? So those are the little tips that you can use. Now let's go to the question. The SA node and AV node are small masses of specialized cells. That is true. AV bundle and Purkinje fibers transmit electrical impulses. Here also from one word, they have made this sentence wrong. It is not AV bundle, it's the AV bundle fibers. Okay, AV bundle gets separated into AV bundle branches. Here they get separated into AV bundle branches over here, and they are the ones which transmit the electrical impulse from the AV node to the apex, not the AV bundle. Okay, so from one word, see how tricky they are. This question is a question where people can get wrong if you didn't know the theory well. AV bundle branches. Okay, now let's go to the third one. The stimulus for contraction of the heart originates in the SA node, yeah, and that is why we call it the pacemaker. Pacemaker, okay, because it originates the contraction. The AV node transmits electrical signals from the atria into the ventricles. Yes, That is the function that is given itself, right? Over here, the AV node transmits electrical signals from the atria into the ventricles. And the fifth one, AV bundle is a mass of specialized fibers. Okay, I don't have to go through this anymore, but let's write down the review since we already found out the answer. Write down the review. The AV bundle branches, and Purkinje fibers, the AV bundle branches and Purkinje fibers transmit electrical impulses from the AV node to the apex of the heart. From the AV node to the apex of the heart. From the AV node to the apex of the heart. The AV bundle branches and Purkinje fibers. Okay, I'm going to stop the class from the 22nd question. I will send you a full recording. Next time, I'm doing a trial session by myself to try to finish the class by 3 hours. So that for the whole month, I'm just taking only 1 day from your month. Okay, 1 day from your month. Now I will tell you how the class goes. I will have to. Okay, hope you can see me. I'm charging my device. That's why I can't keep it upright. I have turned it a bit. So I'm going to tell you what I'm going to do, my class plan and everything. I have discussed only 22 questions. I couldn't discuss all the 50 questions, but I will send you a recording trying to cover up all the questions. My plan is every month I'm going to discuss 3 MCQ papers, right? For the discussion, I will take 3 hours, and the discussion will be not long as this discussion. I will quickly discuss. I decided to give small reviews as well, not to give big reviews. And also I will write down the review in the book itself, so you can take screenshots and then do the review later, right? In the notepad itself, right? And the next thing that I wanted to tell you was, after this is how you should do a paper. Before you start doing the paper, time the paper for 1.5 hours. Do not time the paper for 2 hours, because that's how you increase the speed. Because if you take a paper, I'm pretty sure you do not know all the 50 questions in that paper still. Even though you're studying, even though you have finished the resource book reading perfectly, sometimes you might not know. Like sometimes you might not know how to read this, sometimes you might not know all the 50 questions. You might have forgotten. You might not know the theory of molecular biology, microbiology, applied biology, right? So those questions are there. Ignore those questions if you don't know. That is how you should do a paper when practicing. Do the questions that you know accurately. Even if it's a hard question, but if it's a theory from a known theory, try to do this. Try to take as much as time and do it. But somehow try to finish it with 1.5 hours. Then for the paper writing, I'm advising you to take 1.5 hours. For the paper discussion, I'll take 3 hours. For the review, you have to take 30 minutes. This is how you need to do the review. Since I'm already giving you a review, I'm not going to ask you to write pages. This was my review. See, I wrote pages, right? You can also write reviews if you like, but my advice is, since you're already going for a paper class and if you're planning to join to my paper class, you do not have time to write 2 reviews. And I'm trying to do past papers in this paper class because you do not have a specific other past papers. But tell me, do you like past papers or provincial papers? I'll send you a poll, and I'll send you a poll of the time as well, which time you want me to put the class as well. And also I will first ask, if you want to join to the August month's class, I will put the group link. Those who want can join. And yeah, that's how it goes. So reviewing part, now I'm going to tell you about the reviewing part. Yes. Reviewing part. First, you have to take 3 pens. Don't take color pens. You already have a black pen, blue pen, and a red pen, right? Take those 3 pens. Use red pen for the careless mistakes. Take the paper and circle the ones that you have made careless mistakes. That means the questions in which you have made careless mistakes. When you're reviewing, circle those ones in red. Then the don't know questions. You have some don't know. Molecular biology part, you don't know. So those questions, when you are reviewing it, circle it in black color. and the can't remember questions. You know the cytoplasm very well. You know the plasma membrane very well. But at that time, you couldn't remember the question and you got it wrong. Even if you got it correct, it's okay. If you have got, if you have put a Kana shot or something like that in your book, then definitely you didn't know that, right? Or you knew it, but you didn't, you couldn't remember it, right? So in such cases, if you are going to just answer it in, like, if you answer it in without any sure, without any gut, or you're just answering it because of your gut feeling, then you have to put it into the can't remember section. You can't remember once, you can circle in the blue color pen, right? So that's how you actually separate the questions into the 3 segments: careless, don't know, can't remember, right? Then don't know part, forget it. You don't know that, right? What you have to do is during the week, during the biology study sessions, that is not what I'm going to do. I'm not going to help you with that. You have to do that alone during the biology study sessions. Take your biology book out. Don't study the areas which you already know. What people do is they take the biology book. These are my biology books. See, I have broken down into pages. I have used it so much that they are broken and torn also. So you have to take the biology book. You have to study. You have to keep on studying the areas which you don't know. Molecular biology, microbiology, applied, environmental. Those areas are hard. I will give you some voice recordings in my August month class about the environmental biology, the ones which I used to study, especially molecular biology, DNA parts. I use some recordings while I'm traveling. Okay, there is a low power mode. I think I have to switch off my video. Otherwise, I will tell you more information. I will put a separate video about the class information later. You can watch that, since time is going to get wasted, and also since my battery is low. I'm going to quit the session now. Please give me some comments about the class, whether you like the class, whether you enjoy, like whether you learned something new from the class, whether the class was productive, or whether, if you didn't like the class, honestly say it. If you find anything wrong in the class, anything wrong in whichever sentences I said, tell me, let me know. I am ready for all the comments. So have a nice day. Study well. Study at least for 17 or 18 hours. Now you guys have to study for at least 17 or 18 hours. Take the maximum, use this valuable time for your studies. Okay, do not use social media. Keep a time to use social media. Study hard, study well. Okay, so that's it for now. Have a nice day. Study well. Okay, I will quit the meeting now. Thank you for joining. Thank you very much.